correct preparation strategy of every preparing student for ICAR PG Physical Sciences 2026 should be that he achieves every questions in the examination with successful depth without missing any question from minor portions, right? और AgriAddict का हमेशा से यही मोटो रहा है कि they never compromise with major हो या minor, सारे portions को proper से कवर करेंगे। हमारी मैराथन को हम आज बहुत अच्छी तरीके से minor portions के साथ भी कवर करेंगे और आज का हमारा टॉपिक होगा soil biology and irrigation water quality, classification of soil organisms based on various different categories, and the first classification of soil organisms we have that is on the basis of molecular oxygen. Soil biology, it is very important to understand what is rhizosphere. Rhizosphere is a very important part of soil macroflora, है ना? Plant roots is a very important part of macroflora. Soil microorganisms to be classified into two different categories और very basic categories है, plants है तो soil flora और animals है तो soil fauna. Genus of bacteria which is dominating in every soil system is the Bacillus genus. We are now moving into fungi. Fungi are a eukaryotic, non-photosynthetic, chemoheterotropic group of microorganisms which are playing a key role in mineral tunneling. Actinomycetes are the link between bacteria as well as fungi. They have a similar structural composition of fungi. आप यहाँ पे देख सकते हो कि आपको hyphal kind of structural element दिखने को मिलेगा actinomycetes में, but their characteristic properties, their acting properties are very similar to that of fungi. So they are transition between bacteria and fungi, and they are called as fungi-like bacteria. After completing soil biology, we are now entering into soil quality and irrigation water quality. So hello and welcome again to the YouTube channel of AgriAddict, and here we are back with another session of Maha-Marathon for ICAR PG Soil Sciences 2026 on soil biology and irrigation water quality. So इसके पहले कुछ marathon sessions already पूर्बिता मैम ने ले लिए हैं and रौनक सर ने भी काफी अच्छे से सॉइल केमिस्ट्री और सॉइल फर्टिलिटी को कवर कर लिया है, और जब अभी हमारा सारा मेजर पोर्शन कवर हो गया है तो हमारा मेन इंटेंशन यही होना चाहिए कि हम माइनर्स को भी ना छोड़े, है ना? करेक्ट प्रिपरेशन स्ट्रेटेजी ऑफ एवरी प्रिपरेशन स्टूडेंट, एवरी प्रिपेयरिंग स्टूडेंट फॉर ICAR PG Physical Sciences 2026 should be that he achieves every questions in the examination with successful depth without missing any question from minor portions, right? और AgriAddict का हमेशा से यही मोटो रहा है कि they never compromise with major हो या minor, सारे portions को proper से cover करेंगे, है ना? As we already promised कि marathon sessions में सारे major, minor जितने भी type of questions होंगे most expected series के दौरान आपको बोला गया था कि marathon बहुत अच्छे से cover कराई जाएगी, तो डेफिनेटली हमारी मैराथन को हम आज बहुत अच्छी तरीके से माइनर पोर्शंस के साथ भी कवर करेंगे, और आज का हमारा टॉपिक होगा सॉइल बायोलॉजी एंड इरिगेशन वाटर क्वालिटी. सो बिगिनिंग विद टुडेज सेशन, फर्स्ट ऑफ ऑल आई वुड रिक्वेस्ट जो भी ये वीडियो नए से देख रहा है, एनीवन हु इज न्यू टू आवर यूट्यूब चैनल, anyone who is watching this video first time on AgriAddict's YouTube channel, please click the subscribe button and press the bell icon against it, because we bring so much of quality content for agricultural based exams. And if you like this session, do not forget to share it with your friends and peer mates who are already preparing for ICAR PG Soil Sciences 2026. So ये सेशन आपको आखिरी तक देखना है, क्योंकि we will not just go through the topics, but we will discuss a set of quality questions, brand new set of quality questions authentically prepared by me, which will be very important for exam point of view. So do not miss any part of this session, as this session will be very important. And ये 2 घंटे का, 1.5 या 2 घंटे का जितना भी लंबा सेशन चलेगा, इसमें आपका पूरा सॉइल बायोलॉजी और सॉइल इरिगेशन वाटर क्वालिटी और सॉइल क्वालिटी का पार्ट इतने अच्छे से कवर होगा कि आपको एग्जाम में बिल्कुल दिक्कत नहीं होगी. सो रेडी फॉर महामैराथन? यस. So beginning with today's session, let me briefly introduce myself. I am your mentor Rohit from Soil Science, and I have completed my undergraduation from Anand Niketan College of Agriculture, Anandvan, Varora, which is affiliated to Dr. Punjab Rao Deshmukh Krishi Vidyapeeth, Akola. And currently I am pursuing my masters from Department of Soil Science at Indira Gandhi Krishi Vishwavidyalaya, Raipur, Chhattisgarh. So beginning with soil biology. किसी भी biology का, है ना? Biology is the science that deals with, biology is the science which deals with study of organisms, science which deals with study of organisms. And ultimately soil biology is another part of this study, and hence soil biology is a very important part to be covered. As this part is comprising more and more questions every year, it is very important to discuss this topic in a marathon session. So beginning with soil biology. सोयल बायोलॉजी में इन डेप्थ घुसने से पहले हम बायोलॉजी का एक बेसिक कंसेप्ट, है ना? बायोलॉजी, वन बेसिक कंसेप्ट ऑफ बायोलॉजी इज विटेकर्स 5 किंगडम क्लासिफिकेशन. वी नो दैट विटेकर क्लासिफाइड all the organisms residing on this earth surface into 5 different kingdoms, and these kingdoms are depicted as under. So all the living components classified by Whitaker into 5 kingdom classification as 2 major categories, and all the living classifications, like all the living organisms, are classified into 2 main classes based on their prokaryotic or eukaryotic cellular structure. So what is a prokaryotic cell? Prokaryotic cell is a primitive cellular structure, and eukaryotic cell is an advanced, generally advanced primitive like cellular structure which has a proper nuclear membrane and a proper developed nucleus. So eukaryotic cell is any cell with a proper developed nucleus structure, whereas prokaryotic cell lack the nuclear membrane in the nucleus. So only and only kingdom classified under this prokaryotic organisms are all the single celled monerans, है ना? Bacteria, cyanobacteria and actinomycetes are the all single celled microorganisms which are included under the group of prokaryotic cells, which categorized as living entities, are classified into kingdom Monera by Whitaker. And then he classified eukaryotic organisms into two broad categories, one with single cell with a eukaryotic cell and one with multicellular organization. So whatever the organisms are with single cell and they categorize into eukaryotic class, have a proper nuclear structure, they have a proper functioning nucleus in their cellulotic structure, they are classified into kingdom Protista and they are single celled. So single celled protistans are the transition between these prokaryotic cells and eukaryotic multicellular organisms. Do not get confused in this thing. Okay, so this is very basic, and as 25% of our part of general agriculture may include this all the general biology, we are covering this in depth. So eukaryotic cells are those cells which are multicellular organisms, and they might be somewhere single celled, but mostly multicellular organisms are known as eukaryotes. Then eukaryotes are further classified into three main kingdoms. Three main kingdoms include fungi, plantae and animalia. So kingdom fungi will include all the fungi, rusts, molds, yeast and all the fungal organisms. Then kingdom plantae includes algae, diatoms, desmids, है ना? And kingdoms bryophyta and tracheophyta. Then kingdom animalia includes all the animal structures which comprise of nematodes, worms, insects, centipedes, rodents and all the moles and rodent structures, including us humans, we are included in kingdom animalia. Now apart from this living structures, these five kingdom classification by Whitaker, there is another category which is intermediate between living and non-living, which we call as viruses, and based on the presence of nucleic acid, these viruses are categorized into two different types. One will be viruses associated with RNA and one will be viruses that are associated with DNA. So this is the simple Whitaker's five kingdom classification which can be an important source of question, और हमारा एक भी क्वेश्चन कहीं से भी गलत नहीं होना चाहिए. AgriAddict का हमेशा से यही इंटेंशन रहा है. So होप कि Whitaker's फाइव किंगडम क्लासिफिकेशन सबको क्लियर हो गया होगा. अगर नहीं हुआ है तो वीडियो को पॉज करेंगे, फिर से रिवाइंड करेंगे, फिर से एक बार 10 मिनट की जितनी भी क्लिप थी Whitaker का फाइव किंगडम क्लासिफिकेशन कवर कर लेंगे और आगे बढ़ेंगे, क्योंकि ये एक रिकॉर्डेड सेशन है तो हमारा एक भी पॉइंट सॉइल बायोलॉजी से बिल्कुल भी गलत नहीं होना चाहिए. सो beginning with soil biology. Soil biology is the study of soil microorganisms and some of the important points that are supposed to be remembered, जहां से questions बन सकते हैं, वो points मैं आपके लिए लेकर आया हूं और ये points को बिल्कुल भी भूलना नहीं है. Never compromise with this point, as first unicellular organisms on the earth's surface were known as eubacteriums, है ना? Eubacteriums are the most primitive monerans, है ना? Eubacteriums are the most primitive organisms. Then first photosynthetic cell on the earth's surface. First photosynthetic cell on the earth's surface is known as Gunflintia. Do not forget. Then eukaryotic algae. First eukaryotic algae is Cynidium. First eukaryotic algae is known as Cynidium. Then who categorized or who observed that legumes could utilize nitrogen from atmosphere? पहली बार किसी इंसान ने अगर ये ढूंढा होगा कि legumes can significantly utilize nitrogen from atmosphere is J.B. Boussingault. Okay, and then Bejernik was the first one to isolate root nodules from the legumes. Like root nodules from the legumes for nitrogen fixation were first isolated by Bejernik. इसको भी भूलना नहीं है. These are key discoveries. These are the key discoveries in soil biology, और ये points are very important point for forming questions in any significant exam. And now, when we have different fathers for soil biology, soil physics, soil chemistry, soil pedology. So we have our father for soil microbiology as विंगार्डो बस्की. अब चलो फादर ऑफ सॉइल बायोलॉजी की बात हो ही रही है तो बाकी सारे फादर्स को भी रिवाइज कर लेते हैं. वी नो दैट सॉइल पेडोलॉजी के फादर है जे सेगल. Father of pedology is regarded as Sehgal. Then father of soil physics is Hillel. Then father of soil chemistry is Thomas Way. Then अभी अभी हमने देखा कि father of soil microbiology is Vinogradsky. So these are the important pioneer persons in soil science. And do not forget V.V. Dokuchev. He is the god of soil science. है ना? V.V. Dokuchev is the god of soil science or father of soil science. Then father of soil science in India is also regarded as J.W. Lither. J.W. Lither is regarded as father of soil science in India. Then moving ahead, we are entering into soil classification, like classification of soil organisms based on various different categories. And the first classification of soil organisms we have, that is on the basis of molecular oxygen. So what is the main basic requirement of any living organism? It is oxygen, right? And on the basis of requirement of molecular oxygen into the living structure, the microorganisms have been categorized into three different classes. Those classes are obligate aerobes, facultative anaerobes, and obligate anaerobes. So let's see what are obligate aerobes. Obligate aerobes are those organisms which need oxygen for respiration and cannot survive without it. Obligate there means strictly aerobic organisms, है ना? ऐसे organisms जिनका काम ऑक्सीजन के बिना हो ही नहीं सकता. They need oxygen for their mere survival. ऐसे organisms are known as obligate aerobes. Then facultative anaerobes. Facultative means optional. The word facultative stands for optional. If you see this facultative word, कहीं पे भी अगर ये facultative word आता है तो optional. So optional anaerobe. Optional अगर कोई organism anaerobe है तो वो mainly क्या होगा? Aerobic. So these organisms are primarily aerobic but can survive under anaerobic condition. They have the adaptability to convert themselves into anaerobic organisms. So such organisms which are primarily aerobic but can survive under anaerobic conditions are termed as facultative anaerobic organisms. Then we will see next category. Next category we have on the requirement of molecular oxygen is obligate anaerobes. These organisms cannot survive in the presence of oxygen. They grow in proper absence of oxygen, and such organisms are known as obligate anaerobic organisms. Very simple. Like moving ahead, we have different classification of organisms based on their temperature requirement. So first category we have here are psychrophiles. Psychrophiles are the organisms which survive in low atmospheric temperature. Their minimum temperature can even range up to 0 degrees Celsius, but their optimum ranges from 10 to 15 degrees Celsius. Now what to remember in this table? This table is a AI generated image from the source of treatise, like it is optimized from treatise source. So do not forget to have this table learned as such. अभी मैराथन सेशन में इसका स्क्रीनशॉट ऐसे के ऐसे ले लो, मैं स्लाइड के बाहर खड़ा हूँ, इसका स्क्रीनशॉट ले लो और इसको ऐसे के ऐसे ही रिवाइज करना है. Do not compromise with this table, as this table is a very important source of question formation for the exam. So do not forget, psychrophiles are the organisms which have a temperature minimum requirement, optimum temperature requirement of 10 to 15 degrees Celsius, but their maximum temperature requirement is less than 20. They grow relatively best at low temperatures. Any organism which grows below 20 degrees Celsius is regarded to as psychrophiles. Then there are psychrotrophs. Psychrotrophs and psychrophiles have a mere small difference, है ना? There is a very little difference between psychrophiles and psychrotrophs. Their minimum, see here, psychrophiles have a minimum temperature range of even less than 0 degrees Celsius. They can even survive the temperature range of less than 0 degrees Celsius. Minimum can be less than 0. They can even survive in colder atmospheres. But psychrotrophs have a minimum strict range of 0 degree Celsius. They will survive more than 0 degree Celsius and up to 25 degree Celsius. So their optimum temperature requirement is 50 to 30 degree Celsius. Do not confuse between psychrophiles and psychrotrophs. Psychrotrophs are कोल्ड लविंग ऑर्गेनिजम्स. यहाँ पे आप देख भी सकते हो एक कोल्ड का आइकॉन बना हुआ है. सो साइक्रोफाइल्स आर द ऑर्गेनिजम्स व्हिच लव cold and they grow best at relatively lower temperatures, whereas psychrotrophs are able to grow at low temperature but they refer a moderate range of temperature. So इसको भूलना नहीं है कि they prefer a moderate range of temperature, that is 15 to 30 degree Celsius. Then there are mesophilic organisms. Mesophilic organisms can withstand a temperature up to 45 degrees Celsius, है ना? They can withstand the temperature up to 45 degrees Celsius, but their minimum temperature requirement is 30 to 40 degree Celsius. Most bacteria, है ना? Most bacteria, especially those living in association with warm-blooded animals, like humans, are belonging to this mesophilic category of organisms. Then there are thermophiles. Thermophiles are the organisms which can withstand extensively high degrees of temperature, है ना? अभी गर्मी का सीजन चल रहा है विदर्भ रीजन में और दिल्ली में भी अभी 46 के आसपास टेम्परेचर चले गया था. सो थर्मोफाइल्स आर द ऑर्गेनिजम्स व्हिच कैन विथस्टैंड सच अ टेम्परेचर about 45, like above 45 degrees Celsius and up to 100 degrees Celsius are known as thermophiles. And these thermophilic organisms are very important in the process of composting. बिल्कुल भूलना नहीं है कि अगर composting में कोई organisms important role play कर रहे होंगे तो those organisms are thermophilic organisms. And then there are extreme hyperthermophiles which can withstand temperature as high as 115 degree Celsius. So ऐसे organisms which can survive even the temperature range of higher than 100 degrees Celsius are known as extreme hyperthermophiles. So this was the classification of organisms based on temperature requirement. तो since अब हम उस स्टेज पे आ गए प्रिपरेशन के कि जहां हमें सारा का सारा पढ़ने के बजाय सिर्फ ऑप्टिमम जितना हो स्मार्टली उतना ही पढ़ना है, तो देन वी हैव अनदर क्लासिफिकेशन ऑफ organisms based on pH. Based on pH, there are three main categories: acidophiles, neutrophiles, and alkaliphiles. Very easy with name. We can go with name. Any organisms which can withstand acidic pH conditions, that is less than 6.5 degree, like less than 6.5 of pH, are known as acidophiles. Any organisms which can survive the neutral pH range, normal soil conditions of 6.5 to 7.5 are known as neutrophiles. And alkaliphiles are the conditions which are withstanding temperature range more than 7.5. So, let me correct myself, not temperature, the pH range of 7.5, more than 7.5 and all are called as alkaliphiles. Then acid tolerant ones are also known as oxalophytes. Another name for acid tolerants or acidophiles is known as oxalophytes. Then based on salt content, there are three categories: mild halophiles, moderate halophiles, and extreme halophiles. Mild halophiles are those which can withstand the salt content of 1 to 6%, whereas extremes are the ones which can withstand the salt content of 15 to 30%. Then based on the carbon content, there is an important category, that is oligotrophs and copiotrophs, है ना? So oligotrophs are the organisms with very low nutrient content, and copiotrophs are the organisms which need high carbon content for their survival. Apart from these different categories, there is another important category of rate of multiplication. Rate of multiplication के basis पे soil organisms can be classified into zymogenous and autogenous category. Zymogenous are the ones which have rapid rate of multiplication, and autogenous are the ones which have slow rate of multiplication. So these are the very important categories of soil classification. And now we have most important category, which is very important exam point of view, because questions यहाँ से ही आने वाले हैं, which are photoautotropic, which are chemoautotropic, which are heterotrophic organisms. ऐसे ही organisms आपको exam में पूछे जाने वाले हैं. So let's understand what is this metabolic classification of microorganisms. So first we have nutritional type, let's understand. Nutritionally, organisms are mainly classified into two different categories, phototrophs and heterotrophs. So phototrophs have different categories such as photoautotrophs, then photolithotrophs, photoorganotrophs, then chemoautotrophs, chemoheterotrophs, and chemoorganoheterotrophs. So these are two different classifications, very basic. There is no need to complicate any understanding. First of all, what are photolithotrophs? There is simple understanding कि नाम में ही इसके सब कुछ छुपा हुआ है. So if any organism is photolithoautotrophs, so autotrophs means which can survive, which can significantly create their own food source from the available mineral functions. So photolithotrophs, lithotrophs means they take their carbon from carbon dioxide. Their energy source is light as it is indicated in the name photo, and the electron source, because they are lithoautotrophs, they have an inorganic electron donor. So coming to the photoorganoheterotrophs, their main source of carbon is organic carbon, and since they are heterotrophic mode of nutrition, they take electrons from organic electron donor, but carbon dioxide may also be used. Similarly, here are chemoautotrophs, chemolithoautotrophs. So what will be the carbon source for them? Carbon source is carbon dioxide, whereas energy source is inorganic chemicals, and electron source being inorganic electronic donor. And here are some important examples of these chemolithoautotrophs, such as sulfur oxidizing bacteria, hydrogen oxidizing bacteria, methanogens, and nitrifying bacteria belong to the category of chemolithoautotrophs, which use inorganic chemicals as a source of degradation for their survival. Then chemolithoheterotrophs are the organisms which use organic carbon, but carbon dioxide may also be used, and then they use energy source as inorganic chemicals, and they have inorganic electronic donors, and their example is sulfur oxidizing bacteria such as Beggiota. Then chemoorganoheterotrophs are some of the organisms which use carbon source as organic carbon and energy source as organic chemicals. So this was the classification, and this classification here is simplified into two different categories: photolithotrophs, photoorganotrophs, photoaquatrophs. Here an additional category is mentioned. Do not panic. Just understand that photoaquatrophs is an additional category where just an electron source of water is additionally added. So यहाँ पे भी confuse नहीं होना है. Then there are chemolithotrophs and chemoorganotrophs. So this table is a simplified version of this table. So better you go with this table and then revise the examples based on this table, because it is very easy to understand. Then photolithotrophs, where energy is obtained from light, है ना? First word indicates energy, and the second word then indicates their electron source and carbon source. So this way you can understand the metabolic classification of microorganisms. Then moving ahead, we are coming to rhizospheric root interaction. When studying about soil biology, it is very important to understand what is rhizosphere. Rhizosphere is a very important part of soil macroflora, है ना? Plant roots is a very important part of macroflora. Otherwise आप कहोगे कि सर ने तो macroflora कवर ही नहीं करवाया. अभी बता देता हूँ, rhizosphere is a very important part of macroflora. But since there are many times direct questions seen from this rhizosphere portion, this term rhizosphere was given by Hiltner. ये एक बहुत ही ज़्यादा common objective है. This is a very commonly observed objective in soil science based examination. The term rhizosphere was coined by Hiltner. Sorry. Yes, very good. The term rhizosphere was given by Orenz Hiltner in year 1904. तो अभी क्या कोई मुझे ये बता सकता है कमेंट सेक्शन में कि टर्म फाइलोस्फीयर किसने दी थी, है ना? आई एम वेटिंग. मैं चेक करूंगा ये वीडियो अपलोड होने के बाद कि टर्म फाइलोस्फीयर किसने अपलोड तो कॉइन करी थी. ठीक है, चलो, तो ये क्वेश्चन का जवाब मुझे कमेंट सेक्शन में मिलना चाहिए. ठीक है. देन rhizosphere is a Greek word where rhiza means roots and sphere means surrounding area. So rhizosphere is a interaction between the soil microorganisms and the roots. So what do we understand with rhizosphere? Rhizosphere एक ऐसा जोन है, rhizosphere एक ऐसा जोन है जहां पे रूट्स की मैक्सिमम एक्टिविटी को ऑब्जर्व किया जा रहा है, माइक्रो ऑर्गेनिजम्स की मैक्सिमम एक्टिविटी को ऑब्जर्व किया जा रहा है, एक ऐसा रूट जोन वेयर soil microorganisms are activated and they work very highly, very efficiently. Such a zone is known as rhizosphere. Rhizosphere is further divided into three important zones. First is ectorhizosphere, second is endorhizosphere, and the third zone is rhizoplane. So let's understand simply what are these three different zones into easy categories. So rhizoplane को हम सबसे लास्ट में समझेंगे, पहले समझते हैं कि एक्टो और एंडोराइजोस्फीयर क्या है. लेट्स अंडरस्टैंड व्हाट आर दीज एक्टो एंड एंडोराइजोस्फीरिक जोन्स. सो द एग्जैक्ट बाउंड्री है ना, द बाउंड्री ऑफ रूट्स and the particles, extreme particles in contact with the roots are considered as endorhizosphere. ठीक है. और the ectorhizosphere is the zone apart from 2 mm extending outwards is known as ectorhizosphere. So rhizoplane is the zone between or the plane of contact between ecto and the endorhizosphere, and it extends from roots to 2 mm of distance. ऐसा जोन जो एंडोराइजोस्फीयर से एक्टोराइजोस्फीयर तक एक्सटेंड करे and like approximately 2 mm of zone is known as rhizoplane. तो यहाँ पे देखते हैं rhizoplane, it is a distinct boundary of root surface with the soil and it extends to about 2 mm out from the root surface. Then rhizospheric effect. What is rhizospheric effect? Rhizospheric effect is the effect of extent of enhanced growth of population of microorganisms in the rhizospheric soil. ऐसी soil जहाँ पे plant roots और organisms का एक ऐसा association बन चुका हो and that zone of rhizosphere is having high microbial activity of microorganisms is considered as rhizospheric effect. Then rhizospheric soil in comparison to bulk soil has more population of gram-negative non-spore forming rod-shaped bacteria with simple nutrition requirements. So can anyone tell me कि what are the non-spore forming bacteria और gram negative ही क्यों? ऐसे क्या रीजन होगा कि ग्राम नेगेटिव नॉन स्पोर फॉर्मिंग बैक्टीरिया यहाँ पे हमको देखने को मिल रहे हैं? So the main important characteristic of this non-spore forming rod-shaped bacteria is that हमने commonly observe किया है कि ऐसे बैक्टीरिया है ना, such bacteria which are prone, such microorganisms which are prone, apart from bacteria also, ऐसे microorganisms which are prone to cause any lethal change in plant system, they produce spores, and spores are such a structure which can significantly carry forward the diseased conditions. So major dominating bacteria यहाँ पे कौन से हैं? Non-spore forming, that means beneficial kind of bacteria which are highly involved in nutritional decomposition and nutritional cycling reactions which have simple nutritional requirements. So there are three parts of rhizosphere, endorhizosphere, ectorhizosphere and rhizoplane जिनको हमने अभी देखा था. So since it is a marathon session and none of the parts should be skipped, let me answer this question. The term phylosphere was coined by Ruinen, है ना. अगर ये क्वेश्चन एग्जाम में आ जाता है तो किसी का गलत नहीं होना चाहिए, बट अगर मैं कह रहा हूं कि कमेंट सेक्शन में आंसर आना चाहिए तो एक अच्छा स्टूडेंट कमेंट सेक्शन में आंसर डेफिनेटली करेगा. देन moving forward we have classification of organic materials in the rhizosphere which is chiefly categorized into five different types of organic materials are secreted into this rhizospheric zone, and the first one of it is exudates. Exudates are the low molecular weight compounds that leak from intact cells of plant. ऐसे material which are leaking from the intact plant cells are known as exudates. Exudates मतलब क्या है? Simply अगर words पे जाओगे, exudates मतलब छूटा हुआ, मतलब exclude किया हुआ. So whatever low molecular compounds are leaking from the intact plant cells are known as exudates. So main examples of exudates are amino acids and sugars. Do not forget these important slide. ये slide बहुत ज्यादा important है. वैसे तो आज के सेशन में जितनी भी स्लाइड्स है वो सारी की सारी स्लाइड्स बिल्कुल ही इंपॉर्टेंट होंगी, बट apart from that भी जहाँ पे भी wherever I am telling that this slide is very important, is a very important slide because these are the direct questions, है ना? These questions can be like these exudates, secretion, plant mucilages, म्यूसिजल्स एंड लाइसेट्स आर अ डायरेक्ट सोर्स ऑफ मैच द फॉलोइंग क्वेश्चन. इस पे से मैच द फॉलोइंग क्वेश्चन ऑफ डेफिनेशन और टर्म के मैचिंग का एक्सपेक्ट कर सकते हो. तो इस स्लाइड को बिल्कुल भी कॉम्प्रोमाइज करने की गलती मत करना. सो the most important component secreted by the plant cell excluded low molecular weight compound that is leaking from plant cells are known as exudates. And exudates के बहुत common examples, the most common examples of exudates are amino acids and sugars. Then we have another category which are known as secretions. Secretions are the chemical compounds which are released from plant metabolic processes. Due to the reaction of plant metabolic processes, such secretions are released by plants, and such secretions are examples to be oils, latex, gums and resins. So what are the best examples of secretions? Oils, latex, gums and resins are the well-known examples of secretion. Then we have plant mucilages. Plant mucilages are the secretions, है ना, are the chemical compounds formed due to chemical biodegradation. ऐसे chemical compounds which are released due to plant bacterial biodegradation by plants roots, such a secretions or complex compound produced are called plant mucilages. Then there are mucigels. Mucigels are gelatinous layers composed of mixtures of mucilages and soil particles. अगर कोई जेली सब्सटेंस है ना, देखो कंफ्यूज नहीं होना है म्यूसिलेज और म्यूसिजेल में. म्यूसिलेज are due to bacterial degradation. Any compounds produced by roots due to microbial or bacterial degradation are mucilages, whereas mucigels are the gelatinous secretions of plant roots which comprise of both mucilages and soil particles. Then there are lysates. Lysates are the compounds released by bacterial root cells through bacterial degradation or due to breakdown of or lysis of old epidermal cells. So any substance which is released by plant roots due to breakdown of old epidermal cells, such as substances are known as lysates. ठीक है? Hope this slide is clear and it is very important for exam point of view. Then entering into the main and very important classification of soil microorganisms. First we have soil microorganisms to be classified into two different categories, और very basic categories है. Plants है तो soil flora और animals है तो soil fauna. Soil organisms are basically classified into two main categories. If there are plants, they will be soil flora, and if there are animals like very different microorganisms, earthworms, all rodents, termites, all are classified into soil fauna. So basic classification of soil organisms is into soil flora as well as soil fauna. So soil flora is then classified into two main classes, macroflora as well as microflora. So macroflora अभी हमने देखा था, rhizosphere और roots of higher plants was included into microflora, और जो कि हम अभी बहुत अच्छी तरीके से कवर करके आए हैं. Then we have microflora which include bacteria, fungi, actinomycetes and algae. And then we have macrofauna as well as microfauna. Macrofauna includes earthworms, moles, ants and termites and other rodenticidal organisms which are included under macrofauna, whereas microfauna includes protozoa, rotifers and nematodes. So moving ahead, we have another significant classification directly from ISSS on the basis of size of microorganisms, है ना, on the basis of size of microflora as well as microfauna. Macroflora और macrofauna, इनको sizes के basis पे कैसे compare किया है, here we are observing it. देखो, you will commonly observe that here macroflora category is not included in this classification. Go through this classification. Here macroflora classification is not included, as we know that this is the highest, like maximum size having category, and hence it is not included. Then here we can see that microflora is more in size than microfauna, where microfauna is sized less than 100 micrometer. 100 micrometer of organisms are included under microfaunal category. Then microfaunal category is more in size than mesofauna. Like this is बेस्ड ऑन द नंबर ऑफ लाइक एग्जिस्टिंग बायोमास, कितने नंबर्स में कौन सी कैटेगरी सॉइल में एग्जिस्ट करती है, उसके बेसिस पे इस क्लासिफिकेशन को सीधा सीधा IEEE से उठाया गया है. तो इफ it asks you to arrange into a significant order, do not forget. So we have maximum number of microflora in the soil, followed by microfauna. Microfauna are the organisms with less than 100 micrometer of size. Then we have mesofauna. Mesofauna ranges from 100 micrometer to 2 millimeter. Then followed by macrofauna, macrofauna, and then megafauna. Megafauna is the least number of soil organisms found in the soil. Then on the basis of population we have classifications of flora and fauna. कितनी ज्यादा population किस singular entity की है फ्लोरा में और फोना में, उसके बेसिस पे फ्लोरा और फोना के बहुत ही ज्यादा इंपॉर्टेंट क्लासिफिकेशन दिए गए हैं. एंड दोस टू क्लासिफिकेशंस इंक्लूड बैक्टीरिया बीइंग द maximum in the number. On the basis of number, this classification is on the basis of number, on basis of number or population. The population of bacteria in the soil among flora is highest, followed by actinomycetes, followed by fungi, and followed by algae. So on the basis of population, if somebody asks you, remember the order BAFA. इसमें एक्टिनोमाइसिटीज और अल्गी में बिल्कुल कंफ्यूज नहीं होना है. याद रख लो, अल्गी ऑन द बेसिस ऑफ नंबर हैव द लीस्ट नंबर. सो फॉर पॉपुलेशन इट इज बाफा, and from fauna it is protozoa, followed by nematodes, followed by other organisms and earthworms. So what will be the correct order? P N O E. This is on the basis of population, the number of organisms present in the soil system. Then apart from this, we have another category, dominant organisms based on biomass. Based on biomass, there are two different categories of organisms on flora and fauna. अगर हम देखा जाए कि अभी हमने देखा था कि फ्लोरा का और fauna का क्लासिफिकेशन on the basis of population was BAFA, है ना, बैक्टीरिया were dominating, but on the basis of biomass. अब नंबर और बायोमास में क्या डिफरेंस है? आप बोलोगे कि सर अगर जिसका नंबर ज्यादा है उसका तो बायोमास भी ज्यादा होना चाहिए. नहीं भाई, ऐसा नहीं होता है. ठीक है, समझो कि अगर मैं जहां खड़ा हूं, मेरे उतने ही वॉल्यूम के जगह में इक्वल नंबर ऑफ बैक्टीरियाज अगर भर दिए, है ना, मेरे जितनी जगह में इक्वल वॉल्यूम ऑफ बैक्टीरियाज भर दिए और मुझे भी डिग्रेड किया और वो सारे बैक्टीरियाज को भी डिग्रेड किया, है ना. इफ आई एम स्टैंडिंग हियर, माय वॉल्यूम हैज द सेम नंबर ऑफ बैक्टीरियाज, equal volume of bacteria. If both are degraded simultaneously, which will generate more biomass? Obviously my body will generate more biomass. So me being a singular entity, मैं नंबर में तो एक हूं, बट मेरा बायोमास न्यूमरस बैक्टीरियाज जो साइड में खड़े हैं मेरे ही वॉल्यूम में, उनसे सिग्निफिकेंटली ज्यादा होगा, है ना. Even if me being less in number, the organisms in the same volume of me of bacteria will have lesser biomass. So similarly, there is a case, है ना, on the basis of biomass in floral conditions, fungi dominate in the biomass. So it follows the order Faba, है ना. The third organism on the basis of population comes first when we come to biomass population. So fungi followed by actinomycetes, followed by bacteria and algae. यहाँ पे भी algae का least biomass देखा जा रहा है. Algae have the least biomass in the case of soil microorganisms. Then we have faunal conditions. Faunal conditions here, earthworms. Earthworms देखो जिनकी कम population थी, the ones who were in the least populations are dominating in the case of biomass, है ना. So earthworms followed by protozoa, followed by other organisms and followed by, this is the order for biomass. So hope it is clear, and it should be very very clear and should not be a source of any confusion, because these orders can be directly asked in the exam. So do not get confused. The most dominant organisms in the soil system on the basis of number is bacterial population, and the most dominant floral organism on the basis of biomass is fungi. So moving ahead, we are diving into microfloral population. So microfloral population में हमारी सबसे important category क्या थी? Bacteria. Bacteria because they are most numerous. We know that even a handful of soil, है ना, एक मुट्ठी soil भी हम अगर कहीं से उठाते हैं तो it has about 10 raised to the power 9 times of soil bacteria. तो अगर इतनी पॉपुलेशन है किसी सॉइल बायोमास में, किसी बैक्टीरियल पॉपुलेशन की, तो इट इज वेरी वेरी इंपॉर्टेंट टू स्टडी दीज ऑर्गेनिजम्स इन डेप्थ. सो वी नो दैट बैक्टीरियल पॉपुलेशन में भी वेरियस डिफरेंट टाइप्स ऑफ जीनस है ना. बैक्टीरिया हैव वेरियस डिफरेंट जीनस सच एज बैसिलस, स्यूडोमोनास, देन है ना देयर आर वेरियस डिफरेंट जीनस, थायोबैसिलस, xanthobacter, streptococcus, streptobacillus. There are different genus of bacteria, and among them अगर कोई genus आपको most dominantly देखने को मिलेगा किसी soil में, some genus of bacteria which is dominating in every soil system is the Bacillus genus. Bacillus is the maximum found bacteria in any soil system, which is followed by Pseudomonas in the order of bacterial population. Then we have different shapes of bacteria. If any bacteria has a spherical shape, it is termed as coccus. If any bacteria is having two spheroidal structures connected, it is termed as diplococcus. Diplo means die, die means two, and cocci means spherical structures which are connected, and this is known as diplococcus structure. Then we have streptococci. When the coccus structure form a long chain of cocci, it is known as streptococcus. Then we have bacillus genus, and then we have vibrio. Vibrio are the comma shaped. Then we have spirilla. Spirilla have a tough flagella on one end. Then there are micrococci. Numerous streptococci combine to form micrococci, and then there are spirochetes shape of bacteria. This is very very simple but yet important because this can be a source of match the following questions. Then नॉट वेस्टिंग मच टाइम, क्योंकि ये हमारा ऐसा टाइम पीरियड है कि हमको बैक्टीरिया के जनरल इंफॉर्मेशन में टाइम वेस्ट करने जितना टाइम हमारे पास नहीं है. सो वी विल डायरेक्टली गो इनटू different categories of bacterial population. There are symbiotic nitrogen fixers, non-symbiotic nitrogen fixers, because bacteria play a very important and characteristic role in the fixation of nitrogen. We have seen that nitrogen cycle has very important stages such as nitrification, है ना, aminization, ammonification, and different stages of nitrification are dominated by different processes of bacteria. Bacterial action, bacteria play a key role in the fixation of nitrogen, cycling of nitrogen. Hence it is very very important to study about different groups of soil microbes. So first group of soil bacteria includes symbiotic nitrogen fixing bacteria. So symbiotic nitrogen fixing bacteria are those bacteria, they form close association with plant roots, है ना. With close association with plant roots, they are playing important role in fixation of atmospheric nitrogen, utilization of atmospheric nitrogen with legumes and other plant species which are compatible with the bacterial species. It is very very useful for the fixation of atmospheric nitrogen and conversion of organic form of nitrogen into plant uptakeable inorganic form. So symbiotic nitrogen fixing bacteria include Rhizobium and Bradyrhizobium. Rhizobium has various other cross inoculating groups. For example, Rhizobium japonicum, which inoculates soybean. Similarly, Rhizobium trifolium, which inoculates Trifolium alexandrium or fenugreek. Similarly are the groups of cross-inoculating groups of Rhizobium which can form a close association with various plant forms, and they can be very very effective in fixing symbiotic nitrogen fixation. So there is one single difference between this Rhizobium and Bradyrhizobium, that Rhizobium is a quick, fast and rapid fixer of nitrogen, whereas Bradyrhizobium is a slow nitrogen fixing organism, and it is predominantly found in association with soybean crop. Just keep in mind this thing. So now what is the mechanism of this symbiotic nitrogen fixation? What is the significant mechanism of symbiotic nitrogen fixation? Now let's understand that when plant roots are secreting some sort of substances are secreted into soil system by plant roots such as flavonoids. These flavonoids are the substances which are sensed by these organisms. These rhizobium, bradyrhizobium, various strains of rhizobium and bradyrhizobium have a close association with the plant roots by understanding or sensing that flavonoids group of secretions by plant roots. And whence these flavonoids are sensed by the organisms, they form or secrete a nod C gene, है ना? They secrete a nod C gene, very important gene. Just simply keep in mind, symbiotic nitrogen fixing gene is known as nod C gene for rhizobia, which play a characteristic role in nitrogen fixation. After that, when they sense these flavonoids, they form a close association, है ना? They infect the root with their fiber, and then there is nodule formation, and after nodule formation there is symbiotic nitrogen fixation in the specialized cells of bacterial association. So these are the important genes of symbiotic nitrogen fixing bacteria, rhizobium and bradyrhizobium. And rhizobium has various cross inoculation groups which forms close association with different plants. That should be kept in mind, exam point of view. Then we have another group of microorganisms, that is non-symbiotic nitrogen fixing bacteria. अब अगर कोई symbiotic close association, beneficial association हो सकता है, तो कोई ऐसा भी association हो सकता है जो सिर्फ सिंपली इंडिपेंडेंटली खुद के बेसिस पे किसी और ऑर्गेनिज्म को नाइट्रोजन फिक्स करके दे सकता है, एंड सच ऑर्गेनिजम्स आर नोन एज फ्री लिविंग नाइट्रोजन फिक्सिंग ऑर्गेनिजम्स. फ्री लिविंग नाइट्रोजन फिक्सिंग ऑर्गेनिजम्स, and well known examples of free living nitrogen fixing organisms are Azotobacter, Clostridium and Beijeringia. Do not forget, Beijeringia, Clostridium and Azotobacter are a source of free living non-symbiotic nitrogen fixing bacteria found in soil system. Then we have root associated or associative symbiotic bacterium such as Azospirillum and plant growth promoting rhizobacteria, that are PGPRs. Then we have nitrifying bacteria which converts nitrogen to nitrate, है ना? We have seen that various stages of nitrogen fixation involves different group of microorganisms, and here we see that nitrogen to nitrate is converted by Nitrosomonas, Nitrobacter and Nitrocystis, and these organisms are mainly known as nitrifying bacteria. Then there are ammonifiers which play an active role in ammonification, which include Bacillus and Pseudomonas. Bacillus and Pseudomonas are the two important genus of bacteria which play a key and characteristic role in ammonification of nitrogenous compounds. Then there are denitrifiers. Denitrifiers are the organisms which reduce nitrates into nitrogen gas, and such organisms are well known example of Pseudomonas and Paracoccus. Some genus of Bacillus may also be involved in denitrification. Then there are cellulolytic bacteria. Cellulolytic bacteria which play a key role in breakdown. Lysis, cellulolysis, cellulolytic. The breakdown of cell mass is carried out by cytophaga, sporocytophaga, angiococcus and palliangium. So these are the very important genus of bacteria on the basis of their work they are performing into the soil system. Do not forget, this is a key stage of revision, और यहाँ आपकी revision ऐसी होनी चाहिए कि आप definitely score करो ही करो. So then we are classifying the free living nitrogen fixers into two categories based on nutrition, heterotrophs and autotrophs. But before that, let me take a 2-minute break for water. अभी तक का जितना भी सेशन हुआ आप तब तक रिवाइज करो, and then we will continue ahead. Then on the basis of fixation of nitrogen, free living nitrogenous fixer are classified into two different categories. अभी हमने देखा था कि Beijeringia, Azotobacter ये ऐसे organisms है which fix nitrogen on their own. They do not form any symbiotic association with any plant system, है ना? They fix independently nitrogen in the roots and they convert organic form of nitrogen or plant unavailable form of nitrogen into plant available form, that is inorganic form, and hence these organisms are further classified into heterotrophs and autotrophs. So heterotrophic organisms are further classified on the basis of their requirement of ऑक्सीजन. अभी हमने देखा था वेरियस कैटेगरीज, हमने शुरू ही बेसिक से किया था, है ना, कि व्हाट आर एरोब्स, व्हाट आर माइक्रो एरोब्स, व्हाट आर फैकल्टेटिव एनारोब्स एंड ऑब्लिगेट एनारोब्स. तो वी नो दैट एरोब्स आर द ऑर्गेनिजम्स व्हिच रिक्वायर ऑक्सीजन फॉर देयर कंप्लीट ग्रोथ and activity process. So such organisms which have heterotropic, aerobic as well as free living mode of nitrogen fixation, such organisms are Azotobacter, Dirksia and Bejernikia. Do not forget Azotobacter एक heterotropic, aerobic और free living nitrogen fixing organism है. Then we have microaerobes. Microaerobes are such aerobic organisms which have a temperature requirement, oxygen requirement of less than 0.2 atmosphere, which has a oxygen requirement of less than 0.2 atmosphere. So such organisms are termed as microaerobes. So microaerobes are examples of free living nitrogen fixers of heterotrophic mode of nutrition include Thiobacillus, Methylococcus and Xanthobacter. So Thiobacillus is a very important example of free living heterotrophic microaerophilic nitrogen fixing organisms. Apart from Thiobacillus, Methylococcus and Xanthobacter also include the free living heterotrophic microaerobic population. Then there are facultative anaerobes. What are facultative anaerobes? Facultative anaerobes are those organisms who are primarily aerobes but can survive under anaerobic conditions. So free living heterotrophic facultative anaerobic organisms include Klebsiella, Bacillus and Citrobacter. बिल्कुल भी भूलना नहीं है, दिमाग में फिट बिठा लेना है कि अगर कोई पूछे फ्री लिविंग हेटरोट्रोपिक फैकल्टेटिव एनारोबिक ऑर्गेनिजम्स कोई होगा तो वो है Klebsiella, Bacillus and Citrobacter. Then we have obligate anaerobes, that is they survive in purely absence of oxygen, and such organisms which are free living, they fix nitrogen on their own but have heterotrophic mode of nutrition include Clostridium, Desulfovibrio and Desulfomaculatum. Then we have autotropic और phototropic mode of nutrition consisting organisms. इसमें से कुछ organisms photoautotrophs होंगे, कुछ organisms chemoautotrophs होंगे. So some of the aerobic free living autotrophic organisms include Anabaena, Nostoc and Calothrix, है ना. Anabaena, Nostoc and Calothrix are the examples of aerobic organisms which have autotrophic mode of nutrition, that is they synthesize their own food on their own under free living nitrogen fixing condition. Okay, then we have microaerobic organisms with autotrophic mode of nutrition and free living nitrogen fixation. These are Plectonema, Gliotheca and Lingbaea. We know that Lingbaea, Plectonema and Gliotheca are the organisms which are microaerobic in nature. They have autotrophic mode of nutrition and they are free living nitrogen fixers. Then we have facultative anaerobes as Rhodospirillum and Rhodopseudomonas, and we have obligate anaerobes Chromatium and Thiocystis. Chromatium and Thiocystis को तो बिल्कुल भूलना ही नहीं है. These are autotropic free living but obligate anaerobic organisms. So at this stage of preparation जहां से हमें soil biology का एक एक कोना ऐसे छूना है कि बिल्कुल भी हमारा एक भी क्वेश्चन सॉइल बायोलॉजी से एग्जाम में गलत नहीं होना चाहिए. So the correct answer will be obligate anaerobes asked for autotrophic mode of nutrition including free living nitrogen fixation will include Chromatium and Thiocystis. Then moving ahead, we have some important points to sum up regarding bacteria. अब बैक्टीरिया के इतने सारे क्लासिफिकेशन देख लिए, इतने सारे एग्जाम्पल देख लिए, बट कुछ इंपॉर्टेंट और बहुत ज्यादा इंपॉर्टेंट टॉपिक्स है, कुछ इन्वायरमेंट से कंसर्न क्वेश्चंस आएंगे, है ना. अब एट द ट्रेंड, अब हम उस स्टेज पे पहुंच गए एग्जाम के 2026 एक ऐसा साल होगा जहां पे बिल्कुल भी डायरेक्ट क्वेश्चंस के लिए जगह नहीं होगी. तो हमको इन्वायरमेंट से एसोसिएटेड, है ना, हमारे फिजिकल साइंस डिसिप्लिन में इन्वायरमेंटल साइंस का भी सिलेबस इंक्लूड है. तो हमें हर एक कोने को, हर एक इंफॉर्मेशन को ऐसे कवर करना है कि कोई भी इनडायरेक्ट क्वेश्चन अगर एग्जाम में आए तो हमसे गलत ना हो. और इसीलिए we have some important points to sum up regarding bacteria in association with environment and pollution entities. So presence of enterobacteria is a great ecological significance or indicator of fecal contamination. अगर किसी किसी भी ड्रेनेज सिस्टम में या सॉइल सिस्टम में एंटेरोबैक्टीरिया अगर फाउंड है तो देन इट इज इंडिकेशन ऑफ फीकल कंटामिनेशन, है ना, ड्यू टू कंटामिनेशन ड्यू टू ह्यूमन वेस्ट, बिकॉज एंटेरोबैक्टीरिया मतलब ऐसे बैक्टीरिया विच आर प्राइमरीली फाउंड इन द गट ऑफ ह्यूमंस or gut of microorganisms. If such gut organisms are found in any polluted water source or any polluted soil source or any polluted area or sewage sludge treatment, there we can clearly say that this is the source of fecal contamination. हमको पता चल जाएगा कि इस water source में या water body में या किसी भी soil body में fecal contamination हो रहा है ना। अब फिर उसके वेरियस रीजंस हो सकते हैं, एप्लीकेशंस हो सकते हैं। ये बहुत ही ज्यादा यूजफुल टेक्नोलॉजी है फॉर आइडेंटिफिकेशन ऑफ सोर्सेज ऑफ पोल्यूशन। देन वी हैव बैक्टीरिया बिलोंगिंग टू जेनरा ऑफ डेलोविब्रियो व्हिच आर हाईली मोटाइल बैक्टीरिया। डेलोविब्रियो इज द group of highly motile bacteria. Then there are Myxococcus and Polyandium. These are myxobacteria or slime bacteria, and these play a key role in maintaining equilibrium in soil by feeding on other bacteria. So these are the bacteria which feed on other bacteria. These are myxobacteria or slime bacteria which feed on other bacterial population. Then we have mycoplasmas. Mycoplasmas are the smallest known bacterial population which ranges from the size range of 0.2 to 0.3 micrometers. Do not forget, mycoplasma are the smallest known bacteria with a size range of 0.2 to 0.3 micrometers in diameter. Then we have some bacterial population which use hydrogen sulfide, है ना, which use hydrogen sulfide instead of oxygen as electron donor. अभी हमने देखा था कि these are chemo autotropic organisms, है ना, so lithotropic. So these are examples like green sulfur bacteria, chlorobium, purple sulfur bacteria and chromatium are well known examples of organisms which do not use oxygen as an electron donor source. They use hydrogen sulfide as a source of electron donor. So these were some important bacteria. To sum up point with bacteria, we are now moving into fungi. Fungi are a eukaryotic, non-photosynthetic, chemoheterotropic group of microorganisms which are playing a key role in mineral tunneling. Do not forget this point. Fungi play a key role in mineral tunneling. They are heterotropic organisms. They have a key role in mineral tunneling. Mineral neoformation is the genesis of secondary minerals due to action of soil biota, mostly lichens and mycorrhizal fungi. So mycorrhiza is the association between the roots and the higher plants and lichens is the association between lichens. किस चीज का association है ये मुझे comment section में आप ही लोग बताएंगे, ठीक है। So here we have fungal structure. Fungal structure include fungal fruiting body, है ना, fungal structure include fruiting body as well as mycelium. Mycelium and hypha are the most important parts of fungal anatomy, है ना. This mycelium has a key role in infection and hypha impedes into the any plant system for forming mycorrhizal and lichenic association. So mycorrhizal association are further classified. We know mycorrhizal association is the association between the roots of higher plants and the fungal hyphae. So these mycorrhizal associations are classified into three different categories: endotropic, ectotropic, like endotropic or orchidaceous or non-sheathing type of arbuscular type of association, then ectotropic or sheathing type of association, and ectoendo type of herbaceous type of association. So let's understand what is endotropic and ectotropic. Endo means inside. So any fungal hyphae which is penetrating inside plant roots, है ना, कोई भी अगर fungal hyphae है which is penetrating inside plant roots and it is not forming any sheet on plant structure, then it is known as endotropic type of fungus or mycorrhizal association. And the examples of such association is phoma, है ना. कंसिडर करो कि ये कोई प्लांट रूट है और इसमें फंगल हाइफे ने पेनिट्रेट कर दिया, बट यहाँ कोई शीथ नहीं बनी, यहाँ कोई शीथी स्ट्रक्चर नहीं बना, जस्ट पेनिट्रेशन हो गया एंड देयर इज माइकोराइजल एसोसिएशन, है ना, माइकोराइजल एसोसिएशन व्हिच कैन इवन एक्सटेंड टू राइजोइड्स. तो ऐसा एसोसिएशन अगर बन गया है So such association is known as endotropic orchidaceous. Because why orchidaceous? Because this association is commonly found in orchids. And then non-sheathing type of arbuscular type of association, which has common examples of rhizoctonia or phoma. Then we have ectomycorrhiza. Ectomycorrhiza is a sheathing type जिसमें बाहर एक blanket जैसी layer बन रही है roots के. So such a mycorrhizal association is known as ectorhizal association, and its examples are Boletus and Amantia. Well-known examples are Boletus and Amantia. Then we have ecto-endo-mycorrhizal association or Irrigation type of association. इसके भी example एक बार आप अच्छे से देख लेंगे. Then on the basis of functions, what are the functions of fungi? Fungi are classified into different categories: cellulose and hemicellulose degrading or decomposing fungus, which include an example of Aspergillus, फ्यूजारियम, पेनिसिलियम एंड ट्राइकोडर्मा. ये एग्जांपल्स हमें पता ही होने चाहिए, क्योंकि अगर स्टेटमेंट वाइज क्वेश्चन में ऐसा पूछ ले कि अस्परजिलस इज अ लिग्निन डिकम्पोजिंग फंगस, तो हमारा आंसर क्या होना चाहिए? नो, द स्टेटमेंट इज फॉल्स, बिकॉज वी नो दैट cellulose and hemicellulosic fungus include aspergillus. Then we have lignin decomposing fungus. Many genus of Basidiomycetes group are included in this lignin decomposing fungi. Then we have protein decomposing fungi as Mucor, Aspergillus. See, Aspergillus is commonly included in three different categories, that is humus forming, cellulose and hemicellulose decomposing, and protein decomposing. Now let's understand why it is included in three different categories. Humus forming, that is it has to decompose all the litter and different genera, like different type of compounds found in litter and organic waste. So it is included in protein decomposing as well as cellulose and hemicellulosic decomposing fungi, as it can act on cellulose as well as hemicellulose and lead to degradation and formation of humus substances. So humus forming genera is now including Alternaria and Aspergillus. This should not be forgotten. ये सबके मुंह ज़बानी याद होना चाहिए कि Alternaria and Aspergillus are the two genus of fungi which are primarily included in humus formation. Then moving ahead to the next part, we have actinomycetes. Actinomycetes are the organisms which are important in later stages of composting. ठीक है, actinomycetes are the link between bacteria as well as fungi. They have a similar structural composition of fungi. आप यहाँ पे देख सकते हो कि आपको hyphal kind of structural element दिखने को मिलेगा actinomycetes में, but their characteristic properties, their acting properties are very similar to that of fungi. So they are transition between bacteria and fungi, and they are called as fungi-like bacteria. Then we have number of actinomycetes vary in different kind of soil system. They have a high amount in dry soil as compared to wet soil. Then in grassland and pasture soil, more amount of actinomycetes are found than cultivated soil. Tropical soils have a large population of actinomycetes, and surface soil also have a dominating population of actinomycetes as compared to subsurface soil. Then we know that smell of soil after first rain is very commonly observed due to action of an actinomycetes species, which is commonly known as geosmin. Then these are found in terminal stages of humus formation. ये तो हमें भूलना ही नहीं है कि actinomycetes are very dominant in the later stages of humus formation, as they have the ability to synthesize lignolytic compounds which can degrade lignin, which are high molecular weight compound and which are very dominant in later stages of weathering. They can even withstand anaerobic conditions and higher temperature conditions. So moving ahead, we have another population of soil flora, that is algae. Algae are the chlorophyll containing organisms which have autotrophic mode of nutrition. So soil algae are classified into 4 different types on the basis of their color or the pigmentation. If they have blue-green color, a very common example is BGA. Although it is a bacterial species, it is categorized sometimes as blue-green algae because of its appearance. It is also known as cyanobacteria. है ना? So if it is having blue-green color, it is falling under cyanophyta. If it has grass green or green color, ऐसा color अगर किसी algal population का है, then it is chlorophyta. It is having chlorophyll, है ना? Then xanthophyta are the yellow green colored algal population, and basilerophyta are the golden brown population of algae. Then lichen, अभी हमने पहले स्लाइड में पूछा था कि lichen, what is the lichen? It is the association between the fungi and the algal population, and these are the first to colonize barren rocks. So these play a key role in biological weathering. Since we are studying soil biology, we should not compromise with biological weathering. We know that there is an important role of lichens and roots of higher plants in penetration and action of biota for degradation, breaking, physical breakdown of soil, for formation of soil processes, role of biota, and thus lichen is a very important first colonizer population which play a key role in soil formation. Then ability of blue-green algae to fix atmospheric nitrogen was first observed by PK Day. Do not get confused. Ability of BGA to fix nitrogen was demonstrated by BGA, but agricultural significance of blue-green algae in nitrogen economy was analyzed by RN Singh in paddy soils. अगर RN Singh के बारे में कोई पूछे तो it was in relation to paddy soils. BGA's nitrogen fixing capacity in paddy soils was analyzed by RN Singh. Do not get confused. BGA's ability to nitrogen fixation was given by PK Day. It was demonstrated by PK Day. Then, since we are watching this मैराथन सेशन, अभी ऑलमोस्ट 1 घंटे का टाइम हो चुका है और आप कंटिन्यूअसली इस मैराथन सेशन को वॉच कर रहे हो, तो होप कर रहा हूं, लाइक आई एम होपिंग एज अ मेंटर फ्रॉम सॉइल साइंस कि यू आर एंजॉयिंग अवर मास्टर कोर्सेस एज वेल एज द मॉक टेस्ट बंडल. बट अगर कोई ऐसा है, इफ देयर इज समवन हु हैज नॉट येट परचेस्ड अवर मॉक टेस्ट बंडल एज वेल एज मास्टर कोर्स, you are very kindly requested ki now it is just available for 1999. If you want to achieve the best rankers, as we have already produced from significantly from 3 years, the top rankers are from AgriAddict Academy in ICAR PG exam. So it is humbly requested कि आप इस स्टेज पे भी अगर हमारा मास्टर कोर्स परचेस करते हो तो आपके लिए बहुत बेनिफिशियल होगा, क्योंकि आपको जो भी टॉपिक डिफिकल्ट लग रहा होगा आप सीधा जाके मास्टर कोर्स में वो लेक्चर ओपन कर सकते हो और आप प्रॉपरली, वेरी प्रिसाइसली, गुड क्वालिटी नोट्स के साथ उस टॉपिक को अपने प्रिपरेशन में कवर कर सकते हो और मैक्सिमम मार्क्स स्कोर कर सकते हो. सो मूविंग अहेड, वी हैव प्रोटोजोआ. Protozoans are a very important population of microfauna found in soil, है ना? So these are the predators. The life cycle consists of 2 phases: actively growing phase, that is multiplication, and resting phase, which is forming cyst-like structure in adverse environmental conditions. So protozoa are the dominant organisms or predators which feed on soil bacteria. ये भी हमें याद रखना है. Sometimes this might also be asked. So this is only important point about protozoa. Then we have earthworms. Earthworms are the major है ना, बायोमास के कंसिडरेशन में मैक्सिमम बायोमास वाले माइक्रोफोनल ऑर्गेनिजम्स आर नोन एज earthworms. So the earthworms are the common soil conditioning microorganisms. The worm cast called mull humus is rich in N, P and Ca, which is formed due to the action of earthworms. है ना? Earthworms have some humus forming enzymes in their body which are responsible for breakdown of complex organic substances in the soil. They optimize the soil condition. They develop a fine textured soil. They are more commonly found in fine textured soil than in coarse textured soil. Then vermery is the place where earthworms are cultured, and vermiculture is the process of culturing of earthworms. Then earthworms on their feeding depth, है ना? It is classified into 3 different types. It is a very important classification. This can be asked: what are epizootic earthworms, what are endozoic earthworms, and what are aniseic earthworms? So epizootic earthworms are the earthworms which feed on the soil surface. Such earthworms which feed on the surface litter feeding microorganisms are or earthworms are known as EPG. See, you can pictorially see these type of organisms are feeding on the surface of the earth, and the very common examples of surface feeders are Epinia fetida and Peyonyx excavatus, the most common species of earthworms which are used in vermicomposting. Then we have endozoic earthworms. Endozoic earthworms are the earthworms which feed in horizontal burrows. ऐसे earthworms which are feeding in horizontal burrows are known as endozoic earthworms. And then we have anisaic earthworms. Anisaic earthworms are the earthworms which feed in vertical burrows and are known as deep feeders. They are the organisms which feed in the lowermost zone, deep burial zones of soil. So anisaic earthworms का example होगा Lumbricus terrestica. Then moving ahead to the next slide, we have another important category of macrofaunal population, that is termites. And termites and earthworms are the two main macrofaunal organisms which are known to optimize the physical condition of the soil. So physical modification of soil profile by constructing mounds and foraging runways or tunnels is done by termites. Termites के द्वारा वर्क की गई soils, ऐसी soil which is under the action of termites has high CC and exchangeable bases that surrounds the soil. So termites are cellulose decomposing microbes. They have cellulose decomposing microbes in their guts, and thus their excreta is rich in organic matter, but not more than earthworms. Earthworms ki excreta will have maximum amount of organic matter. So earthworms and termites are known as ecosystem engineers. So now can anyone recall who were known as mineral tunneling organisms? क्या कोई बता सकता है? चलो कमेंट्स में बताना. अभी अभी हमने थोड़ी देर पहले मिनरल टनलिंग माइक्रो ऑर्गेनिजम्स के बारे में पढ़ा है. कौन से ऐसे माइक्रो ऑर्गेनिजम्स थे सॉइल में जो मिनरल टनलिंग में इंपॉर्टेंट रोल प्ले करते हैं? सो अब जबकि हमने सारे सॉइल के मैक्रो, माइक्रो और सारे ऑर्गेनिजम्स कवर कर लिए हैं, तो होप करता हूं कि जितना भी हमने कवर किया है वो सबको इजीली और ऑप्टिमली क्लियर हो गया होगा. अगर नहीं हुआ है तो वीडियो को अभी पॉज करेंगे, बिल्कुल जाके रिवाइंड करेंगे और पूरे सॉइल ऑर्गेनिजम्स के इंपॉर्टेंट कैरेक्टरिस्टिक्स के बारे में देख लेंगे, क्योंकि हमारा टारगेट है आज के महामैराथन के बाद सॉइल बायोलॉजी और सॉइल क्वालिटी को ऑप्टिमली कवर कर लेना. तो जितने भी अभी तक हमने सॉइल माइक्रो ऑर्गेनिजम्स को देखा है, अगर उनके कैरेक्टर्स के बारे में, उनके प्रॉपर्टीज के बारे में, उनके इंपॉर्टेंट एग्जाम्पल्स के बारे में क्लियर हो, तो अभी हम आगे बढ़ेंगे और देखेंगे कि interactions क्या है soil organisms के. Soil organisms have various interactions in within the soil system which result into formation of soil ecosystem. So कुछ ecological interactions के types है which include mutualism and cooperation. Now we know that cohesion है ना, cohesion हमने अभी अभी सॉइल फिजिक्स और सॉइल केमिस्ट्री का पार्ट कवर किया है, तो एडिशन और कोहेजन तो हमें पता ही है. तो कोहेजन, सो व्हाट इज वर्ड को, वेयर इज द वर्ड को इंक्लूडेड? को मींस सिमिलर ऑर्गेनिजम्स. सिमिलर ऑर्गेनिजम्स, तो हम यहाँ पे भी इसको याद रखेंगे कि कोऑपरेशन इज विद इन अ स्पीशीज. बस यही एक की डिफरेंस है कोऑपरेशन और म्यूचुअलिज्म में. म्यूचुअलिज्म इज द म्यूचुअली बेनिफिशियल रिलेशनशिप बिटवीन टू डिफरेंट स्पीशीज. म्यूचुअली एक्सक्लूसिव स्पीशीज. Two different species are mutually benefited by this interaction, and such an interaction is known as mutualism. But cooperation is the interaction between two same species. एक ही species के दो organisms के बीच में जो interaction होगा, it will be termed as cooperation. So mutualism का एक बहुत ही अच्छा example क्या होगा? क्या मुझे कोई बता सकता है? अभी अभी हमने थोड़ी देर पहले देखा है माइक्रोराइजल एसोसिएशन, है ना. माइक्रोराइजल एसोसिएशन इज अ वेल नोन एग्जांपल ऑफ म्यूचुअलिज्म. इसी सब के एग्जांपल्स और ऐसे ही इंटरेक्शन के एग्जांपल्स मैच द फॉलोइंग में बिल्कुल क्वेश्चन फॉर्मिंग पार्ट है, तो इस एग्जांपल्स को बिल्कुल भी एग्जाम के लिए स्किप नहीं करना है. सीधा जाके अगर किसी को इससे ज्यादा पढ़ना होगा तो ISSS खोलना है और इस इंटरेक्शंस के जितने भी एग्जांपल्स है वो सारे एग्जांपल्स को इस चार्ट के बाजू में लिख लेना है आफ्टर मेकिंग नोट्स, राइट. So mutualism and cooperation are the mutually beneficial relationship where both the species are benefited, है ना. बस डिफरेंस इतना है कि म्यूचुअलिज्म में दो अलग अलग स्पीशीज है और कोऑपरेशन में एक ही स्पीशीज के दो ऑर्गेनिजम्स के बीच में कोऑपरेटिव लाइक म्यूचुअली बेनिफिशियल इंटरेक्शन हो रहा है. देन वी हैव कमेंसालिज्म. कमेंसालिज्म इज अ +0 टाइप ऑफ रिलेशनशिप. मतलब ऐसा एसोसिएशन जिसमें एक ऑर्गेनिज्म को तो बेनिफिट हो रहा है बट जो दूसरा ऑर्गेनिज्म है उसका कोई लॉस नहीं हो रहा है, इट इज स्टेइंग न्यूट्रल। जैसे एक स्टूडेंट और टीचर का रिलेशनशिप है, ठीक है, अगर एक स्टूडेंट टीचर का रिलेशनशिप है, एक टीचर स्टूडेंट को पढ़ा रहा है तो बेनिफिट स्टूडेंट का डेफिनेटली होगा ही होगा, बट टीचर के लिए कुछ चेंज हो ना हो उसको तो सारा उसने पढ़ लिया है तब जाके वो टीचर बना है, तो ऐसा रिलेशनशिप हम कंसिडर कर सकते हैं कि कमेंसालिज्म है ना। वन ऑर्गेनिज्म इज बेनिफिटेड from the other without affecting the growth and activity of the later one. And then we have amensalism. Commensalism के बिल्कुल अपोजिट एंटागोनिस्टिक रिलेशनशिप अगर कोई होगा तो होगा अमेंसालिज्म, दैट इज प्लस माइनस रिलेशनशिप, दैट इज किसी एक ऑर्गेनिज्म के वजह से बेनिफिट उठाने के वजह से दूसरे ऑर्गेनिज्म का नुकसान हो रहा है, एंड सच अ रिलेशनशिप इज amensalism. Amensalism is a relationship between organisms of different species, है ना, two different species in which one organism's growth is inhibited and destroyed while the other organism remains unaffected or advantaged. Then we have parasitism or exploitation, where one species parasites or benefits at the expense of host, usually does not killed. अगर host kill नहीं हो रहा है but any parasitic species is invading any host species, it is significantly leading to loss of that host species. Such a interaction is known as parasitic interaction. Then there is interspecific competition, where both the organisms from same species are fighting with each other and leading to a loss of both the species. Such minus interaction between two like same species is known as interspecific interaction, where reduced fitness is observed between two species. Then there is antagonism, the action of any organism that suppresses or interferes the normal growth and activity of the pathogen is known as antagonism. तो antagonism का ही एक example होगा बहुत अच्छा, जो कुछ plant roots secretions secrete करते हैं which inhibits the growth of weeds and microbial population in the soil. ऐसा example will be the example of antagonism. Then there is predation. Predation is when a predator completely feeds and kills the other species, host species is known as predation. So याद रखना है parasitism और predation में यही एक key difference है कि host is getting killed in parasitism, whereas predation में prey is completely killed or consumed by predator. So these are the various set of soil ecological interaction above of soil microorganisms. Then moving ahead, we are diving into soil enzymes, a very crucial part है ना from soil biology, और ये favorite part है, यहाँ से questions अगर नहीं बने तो soil biology में क्या ही पूछ लिया. So beginning with soil enzymes are broadly categorized into two main classes, है ना. हम enzymes की definition फालतू चीजों में नहीं जाएंगे. We know that enzymes are essential compounds which are required for normal functioning and secretions of the body, which are crucially important for the regulation and functioning of normal activity of various microorganisms. So now these enzymes are classified into two different categories: adaptive enzymes and constitutive enzymes. Adaptive enzymes are those enzymes which are formed in relation to selective stimulus. अगर कोई stimulus मिला है और उस stimulus के regard में अगर कोई substrate या enzyme form हो रहा है तो ऐसे एंजाइम को बोलेंगे अडेप्टिव एंजाइम. बट कंस्टिट्यूटिव एंजाइम ऐसा है कि वो कुछ भी हो, प्लांट के नॉर्मल फंक्शनिंग में भी बनना ही बनना है, ऐसे एंजाइम को कंस्टिट्यूटिव एंजाइम कहा जाएगा. तो सम कॉमन इंपॉर्टेंट सॉइल एंजाइम्स आर यूरिएज, डिहाइड्रोजनेज, फॉस्फेटेज, प्रोटीएज एंड अमाइडेज. इनके इन डेप्थ कंपोजिशन में हम नहीं जाएंगे, इनके स्ट्रक्चरल रोल में हम नहीं जाएंगे. हमको बस इस स्टेज पे स्मार्ट प्रिपरेशन करनी है, तो हमें बस इतना याद रखना है कि कौन से एंजाइम का क्या फंक्शन होगा. हाँ, और मैक्स टू मैक्स आप इतना याद रख सकते हो कि कोई भी एंजाइम का इंपॉर्टेंट मिनरल कंपोजिशन क्या है ना, कौन से मिनरल एलिमेंट्स के एग्जिस्टेंस से ये एंजाइम स्ट्रक्चर्स फॉर्म हो रहा है, ये आप याद रख सकते हो क्योंकि वो मिनरल न्यूट्रिशन से भी रिलेट होगा और हमें इनडायरेक्ट रिलेटेड क्वेश्चंस को मिस नहीं करना है. सो वी नो दैट यूरिएज एंजाइम is having an active role in breakdown and volatilization of urea. Then there is dehydrogenase. Dehydrogenase matlab dehydrogenase, that is removal of hydrogen. Removal of hydrogen matlab oxidation. So used to measure carbon metabolism by oxidation of organic matter. Then phosphatase. Phosphatase are the hydrolytic enzymes which act on phosphates by breakdown of phosphate compounds. Then proteases. Proteases have key role in breakdown of proteins. Proteins are converted to amino acid by the action of proteases. Then we have amylases, which are actively playing role in hydrolysis or breakdown of starch. Then there are alpha amylases and beta amylases. Alpha amylases are those which convert starch to glucose and beta amylases are those which convert starch to maltose. बस हमें इतना ही याद रखना है. हाँ, अगर ये भी याद रख लो कि अल्फा अमाइलेज both plants और animals में secrete किए जाते हैं, whereas beta amylase are primarily synthesized into plants. Then there are sulfatase. Sulfatase enzymes are the compounds which lead to breakdown of sulfur rich compounds into sulfate ions. So these are the very important and crucially important things about soil enzymes. Then अब जब हमने soil biology को इतने अच्छे तरीके से पढ़ लिया है तो it is very very important to understand the role of these soil microorganisms in biocontrol, है ना? Control of various diseases, various ill effects of atmosphere are having active role of these soil microorganisms, which is known as biocontrol, है ना? Use of organisms for the control of plant pathogen. So we have an important example of phosphobacterian culture, which is obtained from Acacia occidentalis, है ना? It is an example of phosphobacterian culture which is used in biocontrol of plant pathogen, and it is obtained from Acacia occidentalis. Then we have Agrobacterium tumeficans. ये तो भूलना ही नहीं है भाई. This is very very important. ये previously पूछा भी जा चुका है. Agrobacterium tumeficans can be controlled. The biocontrol of Agrobacterium tumeficans is used by strain 84 of non-pathogenic bacteria which produces antibiotic agrocin 84. Agrocin 84 को यूज किया जा रहा है Agrobacterium tumeficans को कंट्रोल करने के लिए. Then there is siderophore pseudobactin which is produced by Pseudomonas that controls soft rot of potato. Irvinia carnivora का soft rot control करने के लिए siderophore pseudobactin use किया जाता है, which is secreted by Pseudomonas. Then there is Geomyces gramivar tritici, which causes stake-all disease of wheat and barley, which is also controlled. Then Pseudomonas fluorescens is used in prevention of fusarium wilt. Then Bacillus penetrans, which is used to control Melidogyne. Melidogyne is an important species of किस चीज की ये इंपॉर्टेंट स्पीशीज है, आप मुझे कमेंट सेक्शन में बिल्कुल बता देना, ठीक है. देन फंगस नेमेटोफोरा गाइनोफिलिया इज यूज्ड टू कंट्रोल हेटेरोडेरा. These are the some important soil microorganisms which are used in biocontrol of plant pathogens. So moving ahead, we have soil organic matter. अब soil biology पढ़ ली है ना. soil biology अगर पढ़ ली तो soil organic matter के बारे में भी पढ़ना ही होगा. Soil organic matter is the key element which is formed by the action of these soil bacteria, fungi, जितना भी macro, micro, फ्लोरा और फोना है, उन सब के एक्शन के बाद जो बना हमारा soil organic matter, which is of characteristic important, which is of prime importance for our cultivation of crops, cultivation of soil, optimum utilization of organic biomass in the soil और soil की physical health को maintain करने के लिए. तो ऐसा कोई अगर हमारा पार्ट होगा तो वो होगा soil organic matter. तो इसके हमें definition की बात करें तो soil organic matter ऐसा dead, decomposed, highly nutritious mineral matter है which is formed due to action of various microorganisms which have resulted into the decomposition of the litter and the organic waste material which has lead to generation of this soil organic matter. And what is the proportion of organic matter into the soils? ये अगर आपने बता दिया तो आपकी प्रिपरेशन बहुत अच्छी चल रही है. कमेंट सेक्शन में इसका आंसर भी आना ही चाहिए. और अगर बोल रहा हूं ना कि कमेंट सेक्शन में आंसर आना चाहिए तो बिल्कुल आना चाहिए. देख लो, वरना देख लो, मैं कुछ बोल नहीं रहा हूं. अगर नहीं कमेंट्स करोगे ना तो अच्छा नहीं होगा भाई. एग्जाम है तुम्हारे सामने, तुम देख लो, ठीक है. देन देयर आर pools of soil organic matter. Pools of soil organic matter are mainly categorized into two different pools on the basis of their decomposability और breakdown की ability. अगर कोई soil organic matter का pool easily breakdownable है, then such a pool is known as decomposable pool. And अगर कोई pool resistant है, it resists breakdown into simpler substances, then it is known as resistant. So decomposable pool includes metabolic litter and structural litter, where plant and animal residues are the main important components of metabolic litter, which can be decomposed into 1 to 6 months of time period. Whereas structural litters include plant residues like lignin, है ना, branches, ऐसे जो स्ट्रक्चरल कंपोनेंट्स है प्लांट्स के, ऐसे रेसिड्यूज को स्ट्रक्चरल लिटर में इंक्लूड किया जाता है, which needs a comparatively more time for breakdown for about 3 months to 2 years. Then resistant pool includes active, passive and स्लो पूल. रेजिस्टेंट पूल को फर्दर कैटेगराइज किया है 3 डिफरेंट पूल्स में, और इसमें सबसे रेजिस्टेंट अगर कोई पूल होगा तो वो होगा पार्टिकुलेट ऑर्गेनिक मैटर. सबसे एक्टिव पूल अगर कोई है तो लबाइल पूल है, और पैसिव पूल है ह्यूमस. देन दिस टेबल इज डायरेक्टली टेकन फ्रॉम ISSS एंड दिस इज द कैटेगराइजेशन ऑफ सॉइल ऑर्गेनिक मैटर. सॉइल ऑर्गेनिक मैटर को ह्यूमिक सब्सटेंसेस को 3 डिफरेंट पार्ट्स में डिवाइड किया गया है. लाइक सॉइल ऑर्गेनिक मैटर के कंपोनेंट्स को ह्यूमिक, नॉन ह्यूमिक और अदर सब्सटेंसेस में कैटेगराइज किया गया है, where humic substances form the majority proportion. 80% of the part of soil organic matter is from humic substances. Humic substances क्या है ये हम इन डेप्थ देखेंगे ही, उनके प्रॉपर्टीज क्या है ये हम आगे स्लाइड्स में देखने वाले हैं. But then there are non-humic substances such as lipids, carbohydrates and proteins, and other some trace elements are also found. So you can see that 80% और 80 sometimes 85 to 90% even humic substances का प्रमाण या amount बढ़ सकता है, then followed by non-humic and trace substances. So this is about soil organic matter. So then soil organic matter अगर पढ़ लिया तो soil organic matter के degradation में जो कैरेक्टरिस्टिक रोल है, ब्रेकडाउन के रोल में वहां CN रेशियो का भी कुछ इंपॉर्टेंस है, तो CN रेशियो क्या है वो भी पढ़ लेते हैं. द रेशियो बिटवीन अवेलेबल कार्बन एंड नाइट्रोजन इन अ सिस्टम इज नोन एज CN रेशियो. अब हमें ये पता है, अगर सॉइल के इस प्रिपरेशन के स्टेज पे आ गया है बच्चा, तो उसको ये पता होना चाहिए कि जितना वाइड CN रेशियो होगा उतना लेस होगा रेट ऑफ डिकम्पोजिशन, है ना. हायर द CN रेशियो, लेस इज द रेट ऑफ डिकम्पोजिशन. एक इनवर्स रिलेशनशिप है बिटवीन द रेशियो ऑफ कार्बन नाइट्रोजन एंड डिकम्पोजिशन. अगर कार्बन नाइट्रोजन रेशियो कम है तो डिकम्पोजिशन ज्यादा होगा, रेट ऑफ डिकम्पोजिशन ज्यादा होगा, और अगर कार्बन नाइट्रोजन रेशियो हाई है तो रेट ऑफ डिकम्पोजिशन सिग्निफिकेंटली कम होगा. तो हियर यू कैन सी दैट कार्बन नाइट्रोजन रेशियो ऑफ मोर देन 30:1, है ना, मतलब कार्बन नाइट्रोजन के कंपैरिजन में बहुत ज्यादा है. मोर देन 30:1 मतलब अगर 30 कार्बन है तो एक अगर नाइट्रोजन है तो इस केस में इमोबिलाइजेशन होगा. इमोबिलाइजेशन मतलब कन्वर्जन ऑफ inorganic to organic. Then there is mineralization under the conditions of less than 30:1. अगर कार्बन नाइट्रोजन रेशियो लेस देन 30 है, मतलब कि 30 से कम कार्बन है और उसके लिए एक नाइट्रोजन है, तो इस केस में मिनरलाइजेशन की कंडीशंस देखे जाएंगी. और अगर कार्बन नाइट्रोजन रेशियो एट नियर अबाउट 30:1 है तो इक्विलिब्रियम की कंडीशन होगी. साइमल्टेनियसली मिनरलाइजेशन और इमोबिलाइजेशन के प्रोसेस फोरगो करेंगे. देन कार्बन नाइट्रोजन रेशियो फॉर नॉर्मल सॉइल्स रेंज बिटवीन 10 to 12:1 फॉर आइडियल सॉइल, है ना. मोस्ट कॉमनली देखा गया है कि 10:1 दिया जाता है, बट अभी फिलहाल 10 to 12:1 भी कहीं कहीं देखा जा रहा है. तो अगर ऑप्शन में 12:1 भी आया तो बिल्कुल कंफ्यूजन नहीं होने देनी है. कार्बन नाइट्रोजन रेशियो ऑफ एन आइडियल सॉइल इज 10 to 12:1, whereas for sawdust it is 6:1. Then for young leaves it is 10 to 30:1, and for soil microorganisms it ranges from 5 to 10:1, or more precisely 5 to 9:1 for microorganisms, and for cultivated soil it can range up to 8 to 15:1. So for an ideal soil sample it is 10 to 12:1. और ये बहुत सिंपल क्वेश्चंस है, ये अगर एग्जाम में कोई बच्चा गलत करे तो बहुत ही खराब चीज होगी. Then we have extraction of organic matter. Extraction and fracturation इतना important नहीं है, बस इसको इतना याद रख लेना कि it is done with dilute alkali. Dilute alkali सोडियम हाइड्रोक्साइड या सोडियम कार्बोनेट को ट्रीट कराएंगे अगर हम ऑर्गेनिक मैटर से, तो हमें सॉइल के डिफरेंट फ्रैक्शंस, है ना, सॉइल के डिफरेंट फ्रैक्शंस, ह्यूमिक फ्रैक्शंस को एक्सट्रैक्ट कर सकते हैं. अभी हमने देखा था कि सॉइल के 80% पार्ट, सॉइल ऑर्गेनिक मैटर के 80% पार्ट में ह्यूमिक सब्सटेंसेस है और उसके बाद नॉन ह्यूमिक सब्सटेंसेस और कुछ ट्रेस सब्सटेंसेस। तो ह्यूमिक सब्सटेंसेस को एक्सट्रैक्ट करने के लिए देयर आर टू इम्पोर्टेंट, लाइक after we treat it with dilute alkali, then we have to treat it with reflux, methanol और benzene. Then जो भी रेसिड्यू बच जाएगा वो सारे ह्यूमिक सब्सटेंसेस है, और जो सोल्यूबल नॉन ह्यूमिक सब्सटेंसेस होंगे, है ना, जो सोल्यूबल नॉन सब्सटेंसेस होंगे वो मिथेनॉल के साथ निकल जाएंगे। जो बचेगा रेसिड्यू दोस विल बी ह्यूमिक सब्सटेंसेस, और फिर इन ह्यूमिक सब्सटेंसेस को फिर एक बार अल्कली के साथ ट्रीट करेंगे तो हम इनके इनसोल्यूबल और सोल्यूबल फ्रैक्शंस को निकाल सकते हैं। तो हमें पता है कि ह्यूमिन ह्यूमिन इनसोल्यूबल फ्रैक्शन, ह्यूमिन इनसोल्यूबल, ह्यूमिन इनसोल्यूबल, ह्यूमिन, इसको याद ही रख लेना है। देन वी हैव on the basis of solubility in alkali fraction, है ना, there is humic acid and fulvic acid. अगर अल्कली सोल्यूबल कोई है तो वो बोथ है ह्यूमिक एसिड और फल्विक एसिड, और फिर उसके बाद अगर उसको एसिड में ट्रीट करेंगे, वी नो एसिड सोल्यूबल फ्रैक्शन हमारा होगा फल्विक एसिड, लाइक फल्विक एसिड इज कंप्लीटली सोल्यूबल, fully soluble fraction. It is even soluble in alcohol, even soluble in water, even soluble in water, even soluble in acid, and even soluble in alkali. So full fulvic acid is a fully soluble fraction of organic matter, whereas humic acid is only soluble in alkali. So ह्यूमिक एसिड के भी कुछ और कंपोनेंट्स हैं। ह्यूमिक एसिड आफ्टर एक्सट्रैक्शन कैन बी ट्रीटेड विद अल्कोहल टू एक्सट्रैक्ट हाइमेटोमेलानिक एसिड। हाइमेटोमेलानिक एसिड थोड़ा सोल्यूबल कंपोनेंट है, वेयरएज बाकी उसके बचे हुए कुछ ग्रे ह्यूमिक एसिड और ब्राउन ह्यूमिक एसिड कुछ कंपोनेंट्स है जो ह्यूमिक एसिड में पाए जाते हैं। बट हमारे लिए इंपॉर्टेंट क्या है कि ह्यूमिन इनसोल्यूबल है, then humic acid is alkali soluble and fulvic acid is soluble in acid, alkali as well as water. So ये क्लियर है, इसमें कोई भी कंफ्यूजन नहीं होना चाहिए। सोल्यूबिलिटी के बेसिस पे इसमें से अगर कोई भी क्वेश्चन आता है तो बंदे का गलत नहीं होना चाहिए। अभी 1-1.5 घंटा हमने सॉइल बायोलॉजी पढ़ ली है, इसके बाद भी अगर बंदा गलत करके आ रहा है तो कुछ सही नहीं हुआ। Then here we can see that humin is neither soluble in acid nor in alkali, and humin maximum found in vertisols में, then molisols followed by alphisols and spodosols. Then there is humic acid. Humic acid was given by Berzelius in 1830. It was discovered by Berzelius in 1830, but Berzelius is also associated with discovery of proteins, इसको भी याद रख लेना. Then it is soluble in dilute alkali but precipitous on acidification. So it is insoluble in acids. Then fulvic acid, carinic and apocarinic acid are both part of fulvic acid, है ना। एक हमारे मॉक टेस्ट में भी एक क्वेश्चन पूछा गया था कि कॉमन नेम और कन्वेंशनल नेम्स क्या है इन ह्यूमिक सब्सटेंसेस के, तो याद रख लेना है कि ह्यूमिन को अल्मिन भी बोलते हैं, ह्यूमिन को अल्मिन भी बोलते हैं, देन ह्यूमिक एसिड को अल्मिक एसिड भी बोलते हैं। These are the conventional names of this humic substances. So after the humic acid there is fulvic acid which is soluble in both acid and alkali. It dissolves completely in both acid and alkali, and then there are non-humic substances which can be separated by use of benzene. Then there are some important theories of organic matter formation. ऑर्गेनिक मैटर के डिफरेंट थियरीज है, ये किसने दी थी और किस सेंस में दी थी, बस हमें इतना याद रखना है। इससे ज्यादा हमें कोई जरूरत नहीं है जेआरएफ लेवल पे किस बंदे ने कौन सी थ्योरी दी थी और कौन सी थ्योरी कौन से एरिया में इंपॉर्टेंट है, इतना भी याद रख लोगे तो काफी है, क्योंकि हमें स्मार्ट प्रिपरेशन करनी है। हम उस स्टेज पे प्रिपरेशन के पहुंच गए हैं कि जहां हमें भाकड़ पसारे की कोई जरूरत नहीं है, हमें शॉर्ट कंसाइज और ऐसा मटेरियल चाहिए स्पेसिफिक जो एग्जाम में पूछा जाएगा। सो कोनोनोवास थ्योरी वाज गिवन बाय Kononova's humic acid theory. It is important for humic acid. Then Schitzner's theory. Schitzner gave an important theory for fulvic acid. Flake's humus theory is also important. Then lignin theory. Lignin theory is important in swamp areas with poor drainage. इससे डायरेक्ट क्वेश्चन बनते हैं कि lignin theory कौन से एरिया के लिए इंपॉर्टेंट है। Then Stevenson's polyphenol theory is important in forest soils. देखना कंफ्यूज नहीं होना है। lignin theory है ना, आप कंफ्यूज हो सकते हो कि lignin rich substances फॉरेस्ट में गिर लाइक फॉरेस्ट लिटर में फॉरेस्ट में इंपॉर्टेंट होंगे, लेकिन नहीं। स्टीवनसन पॉलीफेनॉल थ्योरी इज इंपॉर्टेंट इन फॉरेस्ट सॉइल। देन मिलियर्ड्स सुगर अमाइन कंडेंसेशन थ्योरी इज इंपॉर्टेंट इन soils with dramatic change systems, है ना। And then Waxman's lignin theory is also important theory of organic matter formation. So these are some important characteristics of humic substances. और ऑर्गेनिक कंपोनेंट्स, इन सबको देख लेंगे। ये स्लाइड का स्क्रीनशॉट ले लो, इसमें एक्सप्लेन करने जैसा कुछ भी नहीं है। अभी हमने जितनी भी चीजें देखी है ना, मॉलिक्यूलर वेट, हाँ बस एक इंपॉर्टेंट चीज याद रख लो कि मॉलिक्यूलर वेट ऑफ ह्यूमन इज हाईएस्ट एंड मॉलिक्यूलर वेट ऑफ फल्विक एसिड इज लीस्ट, है ना। तो इतना अगर याद रख लोगे तो बेनिफिशियल, हाईली बेनिफिशियल होगा हमारे लिए, ठीक है। तो ये कुछ इंपॉर्टेंट प्रॉपर्टीज है, क्रोमोमोर्फिक और प्रोड्यूसिक कंडीशंस। देन यहाँ पे देख लो कि फल्विक एसिड का कलर क्या है, येलो एंड ब्राउनिश कलर है, येलो एंड ब्राउनिश कलर, एंड ह्यूमिक एसिड इज डार्क ब्राउन टू ब्लैक इन कलर एंड ह्यूमिन इज डेफिनेटली ब्लैक इन कलर। तो ये कलर कंपोनेंट भी इंपॉर्टेंट पैरामीटर है, ह्यूमिक एसिड के पैरामीटर्स लाइक ह्यूमिक सब्सटेंसेस के पैरामीटर्स हमें ये पता होने चाहिए। सो कमिंग टू द एंड ऑफ सॉइल बायोलॉजी, है ना, सॉइल बायोलॉजी को कंक्लूड करते हैं, ज्यादा टाइम वेस्ट नहीं करेंगे क्योंकि हर बार 7 से 8 क्वेश्चंस ऑब्जर्व किए जा रहे हैं, तो 2 घंटा, 1.5 घंटा बहुत इनफ टाइम है। तो अभी जितना भी टाइम हमने इन्वेस्ट किया है उसमें हमने डिस्कवर किया कि सॉइल क्या था, सॉइल माइक्रो ऑर्गेनिजम्स क्या थे, फिर सॉइल ऑर्गेनिक मैटर क्या था, सॉइल ह्यूमिक सब्सटेंसेस क्या थे, और अब हम देखेंगे कि ये बॉन्डिंग ऑफ क्ले मिनरल्स, किस नेचर की बॉन्डिंग ये सॉइल ऑर्गेनिक मैटर सॉइल में फॉर्म कर रहा है। तो हम देखेंगे कि मोंटमोरेलिनाइट से बेसल ऑर्गेनिक मैटर लाइक बेसल पोजीशन पे, बेसल पोजीशन, अब बेसल पोजीशन क्या है वो भी समझ लो, है ना। बेसल पोजीशन है ना, बेसल फेसेस पे मोंटमोरेलिनाइट में बॉन्डिंग हो रही है सॉइल ऑर्गेनिक मैटर की। देन इलाइट की बेसल और एज देखो, कोई भी अगर क्रिस्टल मिनरल है तो उसकी एजेस जो है, एजेस पे अगर कोई कंपाउंड बाउंड हो रहा है तो उसको एज नेचर ऑफ बॉन्डिंग कहते हैं। तो बेसल एंड एज बॉन्डिंग फाउंड है इलाइट टाइप ऑफ मिनरल में, और केओलिनाइट जो है वो फेमस है एज टाइप ऑफ मिनरल। तो बेसल बॉन्डिंग इन मोंटमोरेलिनाइट वेयरएज एज बॉन्डिंग इन केओलिनाइट। अगर इतना अभी याद रख लोगे तो काफी होगा। इससे ज्यादा सॉइल बायोलॉजी में अगर कुछ पूछा जाए तो दिक्कत तो नहीं होनी चाहिए, क्योंकि इतना अगर डेप्थ में सॉइल बायोलॉजी अच्छे से 1.5-2 घंटा पढ़ लो तो मोर देन इनफ होगा। मुझे नहीं लगता कि ऐसा कोई भी कॉर्नर छूटा है जो इंपॉर्टेंट है ना। बेसिक तो ऐसा है कि मैराथन में हमको जो सबसे ज्यादा इंपॉर्टेंट है वही कवर करना होगा, सारा का सारा हम लेके नहीं बैठ सकते, लेकिन जितना इंपॉर्टेंट है, वॉट एवर वॉज इंपॉर्टेंट इज बाय नाउ कवर्ड, एंड नाउ वी विल सी सम वेरी इंपॉर्टेंट सेट ऑफ क्वेश्चन सेट फ्रॉम सॉइल बायोलॉजी। तो फर्स्ट क्वेश्चन है हमारे पास, मैच द फॉलोइंग टाइप ऑफ क्वेश्चन। 2 मिनट वीडियो को पॉज करेंगे, अच्छे से क्वेश्चन को देखेंगे, 2 मिनट का टाइम भी नहीं लगना चाहिए क्योंकि हमें 120 क्वेश्चन सॉल्व करने के लिए 120 ही मिनट है। तो बिल्कुल 10 सेकंड में इस क्वेश्चन का आंसर दो। वीडियो पॉज करो, क्वेश्चन को अच्छे से देखो और फिर ही जाके सॉल्यूशन देखना, ठीक है। सो व्हाट इज फर्स्ट, it is asking about father of soil microbiology. Father of soil microbiology is Wingardovsky. Then who first observed that legumes utilize atmospheric nitrogen? Legumes की utilization of atmospheric nitrogen was first observed by Boussingault. Then first isolation of root nodules, ये देखा था हमने कि बेजरनिक ने isolate किए थे. Then first photosynthetic cell on earth surface is Kunflintia. So the correct answer for this question is option B. ठीक है. So moving ahead to the next question, next and very important match the following question. हो सकता है कि ये क्वेश्चन ऐसे का ऐसा एग्जाम में उतर जाए। 2 मिनट लो, वीडियो पॉज करो, वीडियो पॉज करके इसका आंसर खुद अटेम्प्ट करने की कोशिश करो और फिर चेक करो कि तुमने सही अटेम्प्ट किया या नहीं किया, क्योंकि ये सारी चीजें अभी अभी हमने वीडियो में फ्रेशली कवर करी। और अगर इसका आंसर तुम गलत करते हो तो तुम्हारा काम होगा कि तुम फिर से वीडियो को रिवाइंड करो, फिर से एक बार प्ले करो, AgriAddict के YouTube चैनल के व्यूज बढ़ाओ और अपने कंसेप्ट्स क्लियर करो। So we have exudates. What are exudates? Exudates are the compound that are released from the active results of like slowed cells and epidermal cells that are damaged or autolyzed. Then we have wait, ये क्या हो गया भाई? हाँ, so we have mucilages. Mucilages हमें पता है कि बैक्टीरियल डिग्रेडेशन के रिजल्ट से बने थे. Then we have lysates. फर्स्ट ऑफ ऑल एक्सुडेट्स हमारे क्या होंगे, यहां से काट देना और ये क्वेश्चन फिर से स्टार्ट करना, ठीक है। यहां से काट देना और ये क्वेश्चन फिर से स्टार्ट करना, ये क्वेश्चन से हम फिर से वीडियो की शुरुआत करेंगे, ठीक है सर। तो सो अगला क्वेश्चन है हमारा मैच द फॉलोइंग टाइप ऑफ क्वेश्चन, और ये क्वेश्चन ऐसे का एग्जाम में ऐसे का ऐसा उतर सकता है, ठीक है। तो ऐसे का ऐसा उतरने के लिए ये क्वेश्चन देखते हैं कि इसका करेक्ट आंसर क्या होगा, बट बिफोर दैट वीडियो को 2 सेकंड के लिए पॉज करना, इसका करेक्ट आंसर खुद ढूंढना और अगर आंसर गलत करते हो तो वीडियो रिवाइंड करना, फिर से जाना, फिर से पूरा वीडियो अच्छे से देखना, सॉइल बायोलॉजी को अच्छे से कवर करना और फिर जाके इस क्वेश्चन को अटेम्प्ट करना। सो व्हाट आर एक्सुडेट्स? एक्सुडेट्स हमने अभी देखा था कि एक्सुडेट्स आर लो मॉलिक्यूलर वेट कंपाउंड्स, लो मॉलिक्यूलर वेट कंपाउंड्स दैट ooze आउट, है ना। देन फिर है सिक्रीशंस, सिक्रीशंस आर कंपाउंड्स रिलीज्ड ड्यू टू एक्टिव मेटाबॉलिक प्रोसेसेस। सो ए का हो गया सी, बी का डी, देन लाइसेट्स आर द compounds released from dead and autolyzed cells and mucilages are the secretions due to bacterial degradation. So 1 का C, then 2 का D। 1 का C और 2 का D कौन से ऑप्शन में है भाई? एक ही ऑप्शन है ऐसा, अगर इतना भी पता होता। सॉरी 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 सॉरी, C का B। सो इसका करेक्ट आंसर होगा ऑप्शन ए। हाँ, सो C का B है और ये हमने अभी देखा था, सो इसका करेक्ट आंसर होगा ऑप्शन ए। देन मूविंग टू द नेक्स्ट क्वेश्चन, which microorganism is listed as free living nitrogen fixer? तो हमने देखा था कि एजोटोबैक्टर हमारा फ्री लिविंग बट हेटेरोट्रॉपिक मोड ऑफ न्यूट्रिशन होने वाला माइक्रो ऑर्गेनिज्म था। So Azotobacter will be the correct answer for this question. Then earthworms thrive best in soils that are not too acidic, ideally with pH range of, हाँ तो ये पॉइंट को एडिशनली याद रख लेना कि earthworms thrive best in pH conditions of 8.5 to 5.5 to 8.5। ये क्वेश्चन मुझे इंपॉर्टेंट लगा था इसलिए ये मैंने मेंशन कर दिया, ठीक है। देन वी हैव अनदर क्वेश्चन, एन ऑर्गेनिजम्स that strictly require molecular oxygen for its cellular respiration and cannot survive in its absence। वीडियो को 2 मिनट पॉज करो, सही से आंसर करने की कोशिश करो और अगर गलत होता है तो फिर से रिवाइज करो, ठीक है। तो सही आंसर क्या होगा? एन ऑर्गेनिजम्स दैट स्ट्रिक्टली रिक्वायर्स मॉलिक्यूलर ऑक्सीजन फॉर इट्स सेल्युलर ग्रोथ, ऑब्लिगेट एरो, कैन नॉट सर्वाइव इन द अब्सेंस ऑफ ऑक्सीजन। ऐसा ऑर्गेनिज्म होगा हमारा ऑब्लिगेट एरोबिक ऑर्गेनिज्म। तो नाउ जब हमने सॉइल बायोलॉजी को बहुत अच्छी तरीके से कवर कर लिया है, नाउ वी हैव prominently covered soil biology in the earlier 1.5 hour. Now we will be dealing with soil quality and irrigation water quality in very precise way, because these days in the trend last year pretty numerous amount of questions were asked from irrigation water quality and soil quality. It is very important to cover this segment in the मैराथन सेशन, है ना। मैराथन सेशन में हमको ये भी माइनर सेगमेंट बिल्कुल मिस नहीं करना है। एंड एग्रीएडिक्ट बिलीव्स कि ईच एंड एवरी पार्ट ऑफ प्रिपरेशन फेज इज वेरी इंपॉर्टेंट फॉर रैंक फॉर्मिंग फैक्टर्स। फिर चाहे वो हमारा मास्टर बैच का बच्चा हो चाहे ना हो, फिर चाहे वो हमारा यूट्यूब पे देख रहा बच्चा हो, हमारे लिए सारे स्टूडेंट्स सेम है। सो मूविंग अहेड विद द अनदर सेगमेंट ऑफ टुडेज सेक्शन, आफ्टर कंप्लीटिंग सॉइल बायोलॉजी वी आर नाउ entering into soil quality and irrigation water quality. So let's understand what is soil quality. Soil quality is the capacity of specific kind of soil to function within its ecosystem and land use boundary. So now let's understand, if you place any thermophilic organism in a high temperature region, है ना, 80-90 डिग्रीज के टेम्परेचर में किसी थर्मोफिलिक ऑर्गेनिज्म को अगर आप प्लेस कर देते हो, so will that organism will be able to function properly? Yes, because it is a thermophilic organism, it will function optimally in high temperature regions. So soil quality is the capacity. Similarly, in the case of soil quality, let's compare the case of that thermophilic organism. अगर किसी soil को उसके ecosystem में land use boundary में proper functioning की specific kind of functions perform करने की optimal need है, optimally वो soil अगर किसी soil function को within its ecosystem perform कर रही है, proper productivity दे रही है ना, while maintaining the environmental quality and sustaining plant and animal health, then that soil will be called as soil quality. Very theoretical definition, इसमें कुछ बहुत बड़ा साइंस नहीं है, बस इसको इतना याद रखना है कि if at all a soil is properly able to perform its function, that is properly able to produce its yield in its required land use boundary, then that soil is called as qualitative soil. Then what is soil health? Soil health is the dynamic state of equilibrium between the flora and the fauna and its surrounding soil environment. Soil health, when will be a soil called healthy? When the flora and the fauna of any soil system are having equilibrium in their soil environment. At that case, such a soil is known as healthy soil, which is able to perform all the metabolic functions properly and optimally without any hindrance, stress or impedance. If any soil is able to perform its function optimally with high metabolic rate, its optimum metabolic rate, then that soil is called healthy. Now let's compare these soils healthy and soil health and soil quality on the basis of two parameters. Like, what is the major difference between soil health and soil quality? Most prominently, soil health and soil quality are very synonymous term. There is no much difference between soil health and soil quality. But when a soil scientist views it, he calls it soil health, and when an engineer views it, it calls it soil quality. So, what is a soil health basically? Soil health is a parameter that is observed at a state of particular time, whereas soil quality may be observed for usefulness of particular purpose over a long period of time. है ना? At a particular instance, है ना, with a short period of time when it is observed, it is known as soil health, and when it is observed for a long period of time, it is known as soil quality. So now, what is soil health? Soil health is equivalent to the dynamic properties, dynamic small properties which can change over a small period of time, even with a small change in atmospheric and environmental conditions, ecological conditions. If such a conditions can change, it can affect on soil health, and so thus soil health is dependent on dynamic soil property that change in short term. Whereas soil quality is the property which is dependent on the intrinsic or static properties of soil, है ना, which do not change over time, है ना, which do not change easily, which do not get altered easily. Such properties are very much dependent and important for soil quality parameter. So, what are specific soil functions? There are specific soil function, various specific soil function apart from supplying nutrients to the plants. Plant soil is involved in biological diversity maintenance, है ना? There are lots of organisms that are authentically found in the soil atmosphere. Then regulation and partitioning of water and solute flow. We know that water holding capacity of the soil plays an important characteristic role in the recharge of groundwater as well as the infiltration and percolation of activities. That means the water and solute flow is also an important function of soil. Then filtering and buffering capacity and detoxifying and inorganic material storage is an important soil function. Then storing and cycling of nutrients is another important function. Then providing support to socio-economic activities, socio-economic structure and human inhabitation are simple soil functions which were determined by Carlin. Then what is soil degradation? Let's understand. Soil degradation is a merely opposite term or antonym term of soil quality. And there are some physical processes or physical degradative processes which are responsible for the degradation or fall in the quality of any soil. So, soil degradation is the process when there is physical degradation of soil as well as chemical deterioration of the soil biomass. So, it can include high intensity rainfall, then deforestation, urbanization, and various other physical degradative process which can lead to soil degradation. So then moving ahead, we have soil resilience. Soil resilience is a very important property. It is the ability of any soil system to return after disturbance, or it is the ability of a soil to return back to its optimum old and optimal condition which it is supposed to be at. That is, like, consider a spring. If you stretch a spring, है ना, किसी स्प्रिंग को अगर स्ट्रेच कर रहे हो तो अगर ये स्प्रिंग अपनी पोजीशन पे वापस आ पा रही है, इट इज एबल टू अचीव इट्स नॉर्मल कंडीशन, देन इट इज नोन एज रेजिलिएंट, है ना. अगर सॉइल सिस्टम भी कोई ऐसा सॉइल सिस्टम है जिसमें डिस्टर्बेंस होने के बावजूद इट इज ट्राइंग टू गेट बैक टू इट्स ओरिजिनल कंडीशंस, at that stage it will be called as resilient. Soil resilience means the ability of a system to return to its original dynamic equilibrium after a set of disturbances, or ability of soil to resist adverse changes under a given set of ecological land use conditions and to return to its original dynamic equilibrium after the disturbance is known as soil resilience. So there are different types of disturbances. There can be natural disturbances, there can be anthropogenic disturbances. Natural disturbances may include floods, natural floods, tides, etc. whereas anthropogenic disturbances may include tillage or cultural practices. है ना? We disturb the natural normal condition of the soil and hence it is known as disturbance. Disturbance can be called as any significant event that causes a change from the normal functioning of an ecosystem. Then there is soil resistance. Soil resistance is the capacity of soil to function without change, है ना? After any disturbance, कितना भी disturbance अगर किसी soil system पे डाल दो, still it is trying to maintain its optimum pH, है ना? If at all we are increasing the amount of hydrogen ion concentration, still the soil is resisting its pH range, it is maintaining its pH range to its optimum level. At that time, the soil will be said to be resistance. The capacity of soil to continue to function without change throughout disturbance is known as soil resistance. Soil resistance can indicate the degree to which a soil will recover from a particular cropping or management system. है ना? What is the indicator? Soil resistance is indicating कि at what level will the soil recover after particular cropping or management system. Then degree of resistance to change. Degree of resistance to change is the magnitude of decline in the capacity of function. At what magnitude the soil is getting disturbed after disturbance is known as degree of resistance. Then resistance and resilience are two key measures of sustainability and both are dependent on soil type, including soil biota, vegetation, climate and land use disturbances regime. Okay, so moving ahead, we have an important graphical representation of resilience and resistance. See, what do you understand? Degree of resistance is this. The magnitude of change in the optimal soil function is known as degree of resistance. See, first of all, a soil was functioning optimally, but when the disturbance was offered, its functioning capacity was reduced to this 75 here, from 100 to 75, and this 25-degree change, 25% of change in its ability to function is known as its degree of resistance. Then there can be some disturbances and some resilient soil. See, here we are seeing that compaction disturbance after being applied, the soil is returning back again to its normal conditions. So any soil with high resistance will return back to its normal resilient conditions if they are resistant enough. So soils with high resistance after being offered a compaction disturbance returns back to its original normal functioning capacity. But another soil sample, if the compaction disturbance is offered, will suppress down to its functioning capacity to 25% and cannot return back. That soil will be with low resistance. So here another important resistance and disturbance plot you can understand here. When a set of repeated same disturbance are applied again and again. See, if we take 2 or 3 crops in a annual growing season, है ना, एक साल में अगर 2-3 क्रॉप्स लिए जाए तो सॉइल में इंटरकल्चरल ऑपरेशंस लगातार होते रहेंगे एंड द सॉइल डजंट गेट्स टाइम टू रिटर्न बैक टू इट्स ओरिजिनल इक्विलिब्रियम एंड हेंस द फंक्शनिंग कैपेसिटी ऑफ सॉइल डिक्लाइंस विथ टाइम. सो बिफोर, है ना, ऐसा कोई भी डिस्टर्बेंस व्हिच इज ऑकरिंग before its recovery, that will not let soil to recover back to its equilibrium position. Then there are two different parameters of assessment of soil quality, inherent soil quality and डायनामिक सॉइल क्वालिटी. इसमें आप देख लेना, ये स्लाइड का स्क्रीनशॉट ले लो, ये एग्जाम पॉइंट ऑफ व्यू इतना इंपॉर्टेंट नहीं है बट फिर भी इंपॉर्टेंट लगा इसलिए मेंशन कर दिया. There is nothing important in this slide. You can just read the definitions of inherent and dynamic soil quality types. And then moving ahead, we have very important question forming part, soil health scorecard. Soil health scorecard scheme was launched on February 19th, 2015 in Suratgarh, Shri Ganganagar, Rajasthan. Do not forget this. This is very important. Soil health card was launched on 19th February 2015, Suratgarh, Shri Ganganagar, Rajasthan. है ना? And maximum soil health card, soil health scorecards are given by UP government. ये भी याद होना चाहिए. Then tagline of soil health scorecard is स्वस्थ धरा खेत हरा and soil health scorecard must be renewed every 3 years. The population of soil health scorecard, the preparation of soil health scorecard is a qualitative approach. It is a qualitative approach of analyzing the quality of the soil, which depicts both capacity of soil to perform certain functions and different soil attributes, which include structure, soil color, root morphology, earth forms, etc. So moving ahead, there are 3 important rating scales in soil health scorecard. If a soil is healthy, impaired or unhealthy, we can determine it through soil health scorecard. In the scorecard, each soil health indicator is operationalized to confirm the following subject rating scale. That means if a soil is healthy, impaired or unhealthy is determined by the use of or determination of soil health scorecard. Healthy soil means performance of function is optimal and structure is very normal. Impaired means an abnormality in function or structure, or unhealthy soil may be a soil with restriction or inability to perform normal function. Now, there are 12 parameters of soil health scorecard. Which 12 parameters are there? NPK as micronutrients, sulfur as secondary nutrient, only sulfur as secondary nutrient is included in soil health scorecard parameters. Then some micronutrients such as zinc, iron, copper, manganese and boron, and pH, EC and OC as the physical parameters. Do not forget these. These are the very important parameters which are included in soil health scorecard. So इस स्लाइड से एक क्वेश्चन आप डेफिनेटली एक्सपेक्ट कर सकते हो, 4 मार्क आपके इस स्लाइड से बनने ही बनने हैं, तो इसको अच्छे से देख लेना. तो moving ahead we are diving into quality of irrigation water. soil क्वालिटी में इतना ही इंपॉर्टेंट था, इससे ज्यादा कुछ पूछेगा ऐसा तो पॉसिबल होना नहीं चाहिए. अगर पूछता है तो भी आपको डरने की जरूरत नहीं है, क्योंकि हमने मास्टर कोर्स में इससे भी ज्यादा डेप्थ में सॉइल क्वालिटी को अच्छे से कवर किया है. बट सिंस मैराथन सेशन की कुछ लिमिटेड होती है, तो हम मैराथन सेशंस में इतना ज्यादा कंटेंट कवर नहीं करा सकते. बट अगर आपको इससे भी डेप्थ में अगर कंटेंट देखना है, तो आप डेफिनेटली मास्टर कोर्स का सॉइल क्वालिटी और वाटर क्वालिटी का लेक्चर ओपन करो. 1-1.5 घंटे का लेक्चर है, आराम से आपका पूरा सॉइल क्वालिटी और इरिगेशन वाटर क्वालिटी का पार्ट क्लियर हो जाएगा. सो मूविंग इनटू इरिगेशन वाटर क्वालिटी रिफर्स टू द कैरेक्टरिस्टिक्स ऑफ वाटर, है ना. कोई भी वाटर सप्लाई अगर कर रहे हैं हम, इफ वी आर सप्लाइंग एनी वाटर to irrigate our fields, it is very important to analyze the suitability of that water for its specific use, because it should meet the quality parameters. It should not deteriorate our soil. Instead, it should be a supply of mineral nutrients, है ना. अगर सॉल्ट रिच वाटर हम सप्लाई कर देंगे तो हम हमारी सॉइल की फिजिकल हेल्थ कंडीशंस को खराब कर देंगे. इफ वी आर सप्लाइंग अ वाटर दैट इज रिच इन सॉल्ट, it is not useful. It will another parameter to deteriorate physical degradation of the soil. Hence it is very important to analyze the quality of irrigation water. Then soil processes affecting water quality include accelerated soil erosion, leaching, macropore flow and mineralization of humus and microbial byproduct transformation. Then what are the various parameters that are involved in एनालिसिस ऑफ इरिगेशन वाटर क्वालिटी. हमारे लिए बस इतना इंपॉर्टेंट है कि कौन से पैरामीटर्स हम इरिगेशन वाटर क्वालिटी के एनालिसिस के लिए प्रायोरिटाइज करते हैं. और सबसे इंपॉर्टेंट पैरामीटर है हमारा सैलिनिटी हजार्ड और टोटल कॉन्सेंट्रेशन ऑफ सोल्यूबल सॉल्ट्स और द इलेक्ट्रिकल कंडक्टिविटी. देन देयर इज सोडियम हजार्ड, साल्ट इंडेक्स, बाइकार्बोनेट हजार्ड और रेसिडुअल सोडियम कार्बोनेट, और बोरॉन concentration, क्लोराइड concentration, soluble sodium percentage, magnesium hazard, nitrate concentration and lithium concentration. And these most common classification of global level has been given by US Soil Salinity Laboratory by Richards in 1954. But it was given by earlier, it was given by इसके पहले भी अगर किसी ने दिया था वो था विलकॉक्स. तो अगर कोई पूछे कि क्वालिटी ऑफ इरिगेशन वाटर के पैरामीटर्स किसने दिए थे ग्लोबल लेवल पे, यूनिवर्सल लाइक यूएस सॉइल लैबोरेटरी, है ना, यूएस सैलिनिटी लैबोरेटरी, यूएसएसएल ने दिए थे. रिचार्ड्स मेन पायनियर पर्सन था रिचार्ड्स इन 1954. So moving ahead, our first property for analyzing quality of irrigation water is salinity hazard. According to USSL, US Soil Salinity Laboratory classification, 4 salinity classes are there: C1, C2, C3 and C4, which are based on EC. What are the EC values? अगर EC value कुछ है किसी irrigation water की, उसके basis पे irrigation water को 4 classes में divide किया गया है: C1, C2, C3 and C4. C1 class will range from 0 to 25 millisiemen per meter EC, which is suitable for irrigation. Normally irrigation कर सकते हैं. Then 25 to 75 corresponds to class C2, which is suitable for irrigation, but class C3 and C4 ranging from 75 to 225 and 225 to 500 millisiemen per meter of EC are not at all suitable for irrigation water. Normally some amendment practices or some treatment to water is supposed to be applied, and after that application only this water will be suitable for application for irrigation water. Then moving ahead, we have sodium hazard. Sodium hazard is the ratio of sodium concentration to that of concentration of calcium and magnesium, and it is denoted by SAR value. What is the formula of SAR? We know that SAR का formula है sodium adsorption ratio का formula है amount of sodium cations divided by under root of calcium plus magnesium cations divided by 2. So this is the formula of SAR. Huh? And the adjusted SAR was proposed by Soares in year 1981 with general SI unit of SAR as milliequivalent per liter. So on the basis of SAR, soil salinity classes or sodium hazard classes, है ना? Sodium hazard classes are divided into 4 different types: S1, S2, S3 and S4 with SAR value of 0 to 10 corresponding to class S1 where there is no negligible hazard and you can proceed with normal irrigation practices. But if the SAR value is 10 to 18 and it belongs to class S2, there is medium hazard. If the SAR value ranges from 18 to 26, then it corresponds to class S3 and there is high chance of sodium hazard. And if the class corresponds to more than 26 of SAR value, it corresponds to class S4, there is very high irrigation hazard. Then moving ahead, we have salt index. Salt index was a property given by Puri and it is also predicting sodium hazard. The level of hazard sodium is causing is predicted by salt index. It is the another parameter which is dealing with sodium concentration and in relation to sodium, calcium and carbonate ions present in the irrigation water. Then it is the measure of potential of fertilizer to cause salt damage in proximity to roots or seeds. Salt index is the ratio of the increase in the osmotic pressure produced by fertilizer to that produced by the same weight of sodium nitrate, है ना? NaNO3 is taken as a standard value and it has a relative value of salt index of 100. And in comparison to NaNO3, any other fertilizer is predicted to cause that amount of damage to the irrigation water. Now, why it is important? Because many a times we apply irrigation water in the field along with fertilizers, there are chances of damage of roots due to this fertilizer and water interaction. So used to compare change in osmotic pressure when fertilizer was applied in relation to NaNO3. Now salt index value of is negative for irrigation water. If the salt index value is less than 0, then the irrigation water is supposed to be optimum or good for irrigation purposes, and any positive value more than 0, the irrigation water is not at all favorable for irrigating the field. Then nitrogen and potassium fertilizers have high risk of soil damage as compared to P fertilizers. Then another parameter is residual sodium carbonates. Residual sodium carbonates के value है if less than 1.25, it is safely usable. 1.25 to 2.5, it can be used with certain management practices, and if the RSC value exceeds more than 2.5, the irrigation water is not at all suitable for irrigation purposes. Then there is boron concentration. Boron toxicity can be induced due to high concentration of boron, and boron classes are 3 different classes where less than 0.7 ppm of boron concentration crops can grow safely, but if the concentration exceeds 1.5 or more, it is unsafe. So very permissible limits of boron are permitted into the water, and the concentration is important in boron in case because it is in ppm. Okay, so moving ahead, we have chloride concentration. If the chloride concentration in the water is also increasing, then also it is not acceptable. We know we can only use up to 12 milliequivalent per liter of water. That is also slightly usable in case of chloride. So this table is also an important part in question forming era. Then we have soluble sodium percentage and magnesium hazard. Now in soluble sodium percentage, we should understand that soluble sodium percentage value of more than 60 and above is considered harmful for irrigation water. Then magnesium hazard, a harmful effect of soil appears when calcium magnesium ratio becomes less than 50. ये एक ही पैरामीटर है when something less than 50 है ना, less than 50, less than value किसी की good है. Apart from this, another value for this was salt index. Salt index के भी less than value ही ठीक थी, है ना. तो अभी यहाँ पे देखेंगे कि less than 50 value is not good, है ना. The calcium magnesium ratio should be more than 50 if the irrigation water has to be good, and inverse of this is magnesium calcium ratio should not exceed more than 1. है ना. Magnesium hazard in irrigation water is expected when the magnesium calcium ratio is more than 1. So magnesium calcium ratio should be less than 1 and calcium magnesium ratio should be more than 50 for any irrigation water to be optimum. So these are some suitability parameters for irrigation water, and this is the important parameters. These parameters or sequence of suitability of irrigation water have been taken directly from DK Das book, and it is very, very important. Just take a pause at this video, read these properties properly, and just revise sequentially what all properties are important or suitable for the irrigation water application. So in this sequence, chemical composition is having the utmost priority, है ना? pH is having the most priority, then followed by total concentration of soluble salts, then sodium ions, and then the following parameters. So moving ahead, we have some question sets. मैंने पहले ही बोला था वीडियो के शुरुआत में ही कि वीडियो पूरी देखना, बहुत अच्छे से कंसेप्ट्स भी क्लियर कराऊंगा और कंसेप्ट्स क्लियर कराने के बाद क्वेश्चंस भी तुरंत कराऊंगा. सो सिंस अभी बहुत अच्छे से हमने सारा सॉइल वाटर क्वालिटी का पार्ट, सॉइल बायोलॉजी का पार्ट कवर कर लिया है. सॉइल बायोलॉजी के क्वेश्चंस तो पहले ही सॉल्व कर लिए, अभी सॉइल और इरिगेशन वाटर क्वालिटी के भी क्वेश्चंस देखते हैं, बिकॉज दिस इज अ वेरी इंपॉर्टेंट क्वेश्चन फार्मिंग पार्ट. इट इज वेरी इंपॉर्टेंट टू प्रैक्टिस द क्वेश्चंस. वीडियो को 2 सेकंड पॉज करेंगे, पॉज द वीडियो फॉर 2 सेकंड, जस्ट ट्राई टू आंसर द क्वेश्चन एंड देन चेक द करेक्ट आंसर. सो soil health scorecard scheme was launched on February 19, 2015. Yes, we know that. Then residual sodium carbonate index was given by Eaton, salt index concept was given by Puri, and global irrigation standard classification was given by US Soil Salinity Laboratories or Richards in year 1954. देख लेना जो भी correct answer होगा. Then we have next question. Next question is a statement wise question. फिर से वीडियो को पॉज करेंगे, क्वेश्चन को अटेम्प्ट करेंगे और फिर उसके बाद ही करेक्ट आंसर चेक करेंगे. दो ये सेशन रिकॉर्डेड है, बिल्कुल चीटिंग नहीं करनी है. अभी हम उस स्टेज पे प्रिपरेशन के कि जहां पे हमको हमारा बेस्ट देना है, हमको खुद से प्रोमिनेंट रहना है. वी हैव टू बी एक्यूरेट एंड लॉयल एंड पॉजिटिवली सपोर्टिव टुवर्ड्स सेल्फ discipline, right? So at this stage, just pause the video, just try to attempt the correct answer and then check the correct answer for this question. Statement 1 says that the EC classification categorized as C3 class is 75 to 225 millisiemen per meter is designated as highly suitable for irrigation. The statement 1 is wrong. Under the sodium adsorption ratio, hazard rating S1 signifies negligible hazard corresponding to SIR range of value. So the correct answer will be statement 1 is correct, but statement 1 is incorrect, but statement 2 is correct. Then moving ahead, we have another match the following question based on definitions and their important parameters. So here a word is missing, soil quality. Just put the word soil quality here. So soil health, we know that soil quality is the ability of soil, capacity of soil to sustain biological productivity and maintain environmental quality, whereas soil health is the state of dynamic equilibrium between flora and fauna. Then soil resilience is the ability of soil to return to its dynamic equilibrium after disturbance, and soil resistance is the capacity of soil to continue to function without any change or damage. So whatever will be the correct answer, 1 ka B, then 2 ka A, 1 ka B, 2 ka A, 3 ka D and 4 ka C. So the correct answer will be option A. Then moving ahead to the next question. Next question is another statement based question based on soil health scorecard. Soil health scorecard पे क्वेश्चन है, वीडियो को पॉज करेंगे, 2 मिनट में क्वेश्चन अटेम्प्ट करेंगे. जस्ट पॉज द वीडियो, जस्ट ट्राई टू अटेम्प्ट द क्वेश्चन ऑन योर ओन एंड देन check the correct answer. So statement 1 says that it was launched on 19 February 2015 with the official tagline of Swasthadhara Khetara. Yes, the first statement is absolutely correct. Then statement 2 says that soil health scorecard must be renewed by farmers after every 5 years. What do you think will be the correct answer? The card monitors a total of 12 parameters which include secondary nutrients like sulfur and physical parameters like organic carbon. Yes, this also statement is correct. So now just drop the correct answer for this question in the comment section, क्योंकि हमारा सेशन बिल्कुल इंटरैक्टिव होना चाहिए. आप अगर कमेंट करोगे तभी मुझे पता चलेगा कि आपने ये सेशन सही से देखा है या जस्ट स्किप कर दिया है. So just drop the correct answer in the question. Like then we have another match the following question. Just वीडियो को पॉज करो, क्वेश्चन अटेम्प्ट करो. जस्ट पॉज द वीडियो फॉर 2 seconds, क्वेश्चन अटेम्प्ट करो, कोई भी चीटिंग नहीं करनी है and then check the correct answer. Then global USSL classification was given by Richards in 1954. देखो यहाँ पे भी USSL mentioned है और यहाँ पे भी USSL, तो आपको एक आंसर तो क्लियरली यहीं पे मिल गया. Then salt index was given by Puri, residual sodium carbonate by Eaton, है ना, and adjusted SAR by Saurus. So the correct answer will be 1 का C, 2 का A, then 3 का D. So the correct answer will be option B. Then moving ahead to the next question, what is the capacity of soil system to continue functioning without any change or disturbance? It is known as soil disturbance. To continue without any change is known as soil resistance. Yes. Then moving ahead to the next question. Which parameter group includes the physical indicators assessed on soil health scorecard? So what is the correct answer for this question? Physical indicators include pH, EC and OC in soil health scorecard, whereas micronutrients like molybdenum, iron, zinc, है ना, and manganese, then secondary nutrients like sulfur, primary nutrients like NPK. कितने parameters हो गए भाई? 1 2 3 4 5 6 7 8 9 10 11, है ना. Iron, molybdenum, zinc, manganese and copper, I guess, is the another micronutrient which are the parameters of soil health scorecard. So this was about today's session. In today's session, marathon session में बहुत ही अच्छी तरीके से soil biology, सॉइल क्वालिटी और इरिगेशन वाटर क्वालिटी को भी कवर कराया है और उससे एसोसिएटेड जो कुछ एक्सपेक्टेड अच्छे क्वालिटी के क्वेश्चंस है वो भी कवर कराए गए हैं. अगर आपको ये मैराथन सेशन बेनिफिशियल लगे हो और ऐसा लग रहा है कि मैराथन के अलावा भी आपको ज्यादा पढ़ना है, तो इट इज हाईली रिकमेंडेड कि नाउ अभी भी टाइम है, अभी भी पूरे 20-25 दिन का टाइम है. जस्ट गो एंड बाय द मास्टर कोर्स फॉर 1999 and cover the concepts in depth. It will be very beneficial for the exam point of view. And if somebody is watching this session for 2027 master course, it is highly recommended to join the Shrestha batch. Shrestha batch is now being launched and it is now available for 4,999. It will include all the live classes, all mock tests, their explanations, proper notes, high quality questions and everything which is required for ICAR PG exam. Hope you enjoyed this session. Hope this session was beneficial for each one of you. Will expect पॉजिटिव एज वेल एज नेगेटिव कमेंट्स इन द कमेंट सेक्शन. एंड अगर ये आपको सेशन पसंद आया हो तो इस वीडियो को लाइक करें, सब्सक्राइब करें और हमारे चैनल को एज फार एज पॉसिबल ज्यादा से ज्यादा शेयर करें और सभी के लिए बेनिफिशियल बनाएं. थैंक यू फॉर ज्वाइनिंग टुडेज सेशन. थैंक यू फॉर ट्रस्टिंग एग्रीएडिक्ट. थैंक यू.